Okay. Hi, everyone. I'm excited to be here, and I'm honored to present to you my research on tip-growing cells and their shapes, specifically how they generate their shapes and why they have the shapes that they do. Okay, so to begin to understand this question, first let's start broadly by thinking about cell shape. So how is cell shape determined in nature? So there's all sorts of shapes that cells can have in nature. They can have this sort of intricate geometry, such as microsterias. They can be cylindrical, like in bacteria, or they can just be amorphous blobs, like many of you might be familiar with mammalian cells. So there's a wide diversity of shape, and the question is, how is this determined? And to begin thinking about it, let's first think about the sets of possible morphologies, of the sets of cell shapes. So there's a set of conceivable shapes, sort of what is conceivable. And we can constrain this by thinking about a specific morphogenic process, for example, that is unique to a given cell or given that certain cells use. And so the thing about morphogenic process is that it's developmentally constrained. So there are only so many shapes that are developmentally possible given a certain morphogenic process. And then but there are certain shapes that are possible. However, cells are biological entities that are subject to the laws of evolution and natural selection. So not all the shapes that are developmentally possible are useful or advantageous. So the shape of cells is further constrained by selection for evolution. And this is the set of shapes that we observe in nature. So for this talk, I'll tell you about tip growth. And tip growth is a morphogenic process in walled cells. So cells with so this applies to cells that have a cell wall, which is a cross-linked polymeric structure that's fairly solid and encases the cell. And in tip-growing cells, they exhibit a localized cell wall synthesis at an apical point that results in this highly dynamic, highly polarized morphogenic process that is self-similar. Tip growth is found all over the tree of life. For example, it occurs in all sorts of vegetatively growing organisms. For example, it can be found in pollen tubes, which are in plants. And a pollen tube is essentially the way plants are able to deliver the male gamete to the female gamete in a flower. And so it's important for their reproduction. Tip growth has also evolved in fungal hyphae. So hyphae are tip-growing cells, and this is a way that fungi explore the environment for nutrients and resources using growth. And something that I find extremely interesting is that there are also these protist species that have evolved tip-growing cells that look just like hyphae and are similarly used for exploring the environment for nutrients and resources. But also some of these species are parasitic and they use this to invade their hosts. All right, so for this talk, I'll tell you how, I'll convince you that tip growth is an ideal system to study cell shape morphogenesis. One, because they have a common morphogenic process that we can define for them. And two, they have some variability in apical morphology that ranges from round to tapering. And this got us asking a few questions. Essentially, how is variability in apical morphology generated? And why do tip-growing cells have the shapes that they do, specifically? So a quick overview of what you should expect from this talk. So we will model tip growth mechanically as inflated cell wall growth. And by modeling it in this way, we're able to understand that we're able to extract a profile of surface extensibility. And we're able to understand that this is what modulates apical morphology. Using surface extensibility, we're able to simulate the entire morphospace, theoretical morphospace of possible apical morphologies in tip-growing cells. And that morphospace tells us that naturally occurring cells are optimized have shapes that optimize for fast elongation rates. And we also discover a cusp bifurcation in the more in the morphospace that constrains apical morphology. All right, to begin thinking about this first, let's characterize, we can characterize the apical tip mechanically. And to do that, we can define two principal axes of orientation. We can define a meridional axis, which is along the apical tip, and then a azimuthal axis, which is around the circumference of the tip. And we can measure certain quantities, principally the cell wall expansion rates or the strain rates, which quantify the rate of deformation of the cell wall along this apical tip, and the cell wall tensions that arise due to pressure inside the cells, which also vary based on the apical geometry. And so using this framework, we're able to define a model in which the cell wall expansion rates are linearly related to the cell wall tensions via the cell wall mechanical properties, whereby the gamma here is a surface extensibility or an inverse viscosity and quantifies how easily the cell wall flows along the apical tip. And another parameter is the flow coupling, which quantifies the coupling between perpendicular expansion rates, but for the purpose of this talk is not a very important parameter for understanding cell shape and is assumed to be a free parameter with constant value. All right, so something I want to kind of drive here is that surface extensibility varies spatially along the apical tip meridian. We can define it as a spatially varying variable. I'm focusing on surface extensibility, but actually also these other variables are spatially varying along the meridian. Okay. So we were able to empirically derive surface extensibilities for 3 species. We chose these species because they span morphology. So they range from the highly tapered morphology to a highly rounded morphology, but they also span biological diversity, whereby each of these 3 organisms is from a completely different kingdom. This is a plant, so it's a pollen tip, this is a fungus, and then this is a protist. And so they share no sort of evolutionary homology, they just convergently evolved tip growth. And so what we can do is we can measure the wall expansion rates and the wall tensions for these 3 species experimentally. I will not explain that now, but you can ask me later if you're curious. And given that these are measurable quantities, we can empirically derive a surface extensibility for all these 3 species, which looks like this. So let me explain this plot. So the x-axis is the arc length, which is the distance away from the pole. So S equals 0 is this point at the apicalmost point. And as you move negatively or positively away from the pole, you increase the arc length. And then the y-axis is the empirically derived surface extensibility. And as you see, for all these 3 species, the surface extensibility is maximal at the pole, which is kind of intuitive because the tip growth, so they're growing from their tip, so they would be most pliable. The cell will grow and be most pliable at that apical point. And then the profiles decay in sort of characteristic ways for these 3 species. Yes.
The active element here is the tension.
Yes, the tension drives. So this is inflation. We're modeling as inflation-driven growth, whereby the cell wall tensions are driving the cell wall expansion.
The response of the cell wall is completely passive.
In this case, yes.
And is there an intuitive argument for why that should be true, that you can make this separation the only active element of tension? Nothing about how the wall responds, which is active. It's not coupled to internal machinery.
Okay. So I think an intuition comes from the fact that all the active processes in the cell, in this cell, are really only happening here. Okay. Yes. And then these cells also, I think maybe it's not clear, is Since these cells have very high turgor pressure, like significantly higher than mammalian cells or any cell that doesn't have a cell wall. They're really, really high. And they're sort of the dominant source of force in these cells, much more than the cytoskeleton or sort of insertion-based mechanisms of growth.
This may be a basic question, but did we start from, did we assume at any point that like the cell is elastic or viscoelastic material?
Yes, there's actually an implicit assumption of viscoelasticity.
Okay.
Yes, that's a viscoelastic material that flows more easily at the apical plane. It's precisely that.
Thank you.
Mm-hmm. Okay. All right. So something extremely interesting is that now that we have this empirical surface extensibility profile for three species, and we have this linear conceived model that relates these quantities, we developed a numerical simulation whereby all we input is this experimental surface extensibilities and define everything else using a geometrical initial condition, and then run the simulation to convergence until a stable like shape is obtained. And what happens is that just by knowing this empirical surface extensibilities, you're able to recapitulate the experimental shapes exactly, precisely. And so this tells us that the surface extensibility is sort of the key parameter of interest for understanding how a tip-growing cell modulates its morphology. And so when we pay attention a bit more to these extensibility profiles and the characteristic way in which they decay, what you'll notice is that for the non-rounded shapes, there are actually two length scales over which this extensibility decays, whereby, for example, this highly tapered one, where it's really obvious, the extensibility decays rapidly over this short length scale, and then abruptly it starts decaying rather slowly over a much longer length scale. And it's so to a less dramatic effect for this mildly tapered hypha cell. And for the rounded pollen tube, it essentially decays over one length scale. Yes.
Maybe it's not a good question, but just in case, you suggest that what drives the tip shape and the tip growth is mechanical pressure?
Yes.
Rather than, for example, I could think about another alternative, which would be like a programmed process of the underlying cell and gene regulatory network. So that's just to understand, like, you suggested the driving force for the way the tip is shaped and the way it's driven forward and getting longer is purely due to mechanical stress and not to like program
cell regulation.
Yeah.
So the cell, of course, can regulate itself. But what I'm suggesting is that it's the mechanical expansion is the main driver. It's the dominant factor determining all this. So genetically, they might determine how the rate of synthesis and sort of how they modulate that, like the properties of the cell wall. There are many complex ways they can biologically like control themselves. But the key insight here is that the mechanical forces are dominant in this morphogenesis.
And will you explain soon? I mean, I can see the associated relation, but it's hard for me to capture like the causal relation. Can you explain? Will you explain it later? How do you conclude that the mechanical driven force that's the dominant force rather than like a...
Yes, that's a good question. Experimental data that shows this.
Okay.
I have a slide.
Yeah.
And I can show you.
So it's a good question.
Yeah. So I didn't explain it for this talk because it's a bit long-winded, but we know like there's a very this data that shows this. This is empirical. Yeah. Okay. So remember I told you that the extensibility decays over two distinct length scales. So what we did is we attempted to fit these extensibility profiles using a function that can capture these two length scales. And in this case, we use the function that we chose. It's called the Gaussian mixture. It doesn't have to be this, but this is the most convenient to use. And essentially, the Gaussian mixture is a combination of two Gaussian curves one that captures the height of the extensibility profile and one that captures the width of the tails. And there are two parameters that are important for characterizing this function, which is the ARAT, the ratio of the amplitudes of the peaks, and the LRAT, the ratio of the length scales of the peaks. And so we can just kind of, find parameters that fit well. And this function is able to fit the Gaussian the extensibility profiles precisely or very, very well. And in this sense, remember how I told you we have this simulation and that we can just input an extensibility and with some initial condition, it gives us a shape. So what we can do is we can just input these Gaussian fits, these Gaussian mixture fits into the simulation routine. And lo and behold, this is enough to recapitulate the apical morphology very well. Not perfectly, but very well. Yes?
Is something that is regulated or needs to be regulated by the cell, or is it natural that it comes out this way, or that it moves with the tip, or is it something that is?
It could be generally what we've observed in like cells is, or like some experimental data from some papers on different cells over the literature, is that it's regulated. That they have enzymes that soften the cell wall at the tip, and then they have cross-linking enzymes that stiffen the cell wall further back. So they're able to kind of set this. Yeah, it's highly dynamically regulated. It's a whole process of like synthesis and cell wall remodeling and stiffening, and it's very dynamic. If you really look into the cell biology of it precisely. Okay, so the point I'm trying to make with this Gaussian mixture function is that it's actually a really powerful function for modeling surface.
Very simple question. The plot in the middle is an order of magnitude smaller than the one on the sides, like 10 to the minus 3, 10 to the minus 4.
Yes.
There is any reason why that should be the surface extensibility is like one order of magnitude smaller in the middle case?
I think it's a species-specific property. It's specific to the species.
Okay.
Yeah. Because we empirically derive from the expansion rates, and like it has certain expansion rates.
So the magnitude doesn't matter. Like
yeah.
the magnitude just let the shape of the curve.
Yeah, the magnitude doesn't matter. Like it still will give you the same shape. Maybe it'll just be faster growing if the extensibility is higher.
Okay.
Yeah. If the magnitude just changes, maybe it relates to the expansion rates. Okay. So this Gaussian mixture function is really powerful because it lets us model surface extensibility in a very highly controllable way. So what you can do is that we don't only need to, you can do much more than just fitting this profile. We can actually define hypothetical extensibility profiles and then simulate those. And this is precisely what we did. So we generated a phase space of extensibility profiles, whereby using the parameters of the Gaussian mixture, and whereby the y-axis here is the ARAT, the ratio of the amplitudes of the two peaks, and LRAT is the ratio of the length scales between the two peaks. And each point here corresponds to a unique extensibility profile that uses two length scales, that is defined by two length scales in some way. And then we simulate the shapes that they converge to, and we can quantify the shapes. For the purposes of, I'll just say we use the PCA. I can explain to you if you're curious later how this works, but we use a PCA to morphometrically quantify the shapes. And it outputs a number that it outputs a principal, the first principal component that gives us a number of basically how rounded versus tapered the shape is that is output. And so when we overlay the values of the P1 of the principal component here, the PCA analysis here, morphometric PCA analysis, what we get is a morphospace, okay? Whereby, given sort of all these extensibility profiles and what we could simulate, we generate most of the shapes that are simulated seem to be quite round, and only a small set of parameters gives you highly tapered morphology, where this diamond is. However, the shapes in this sort of rounded regimes are not the same. And so again, this PCA also has a second principal component, and basically the values of PC2 are different. PC2 captures sort of fine scale changes in curvature. And okay, so to kind of interpret what's going on here, what we just did is that we simulated the range of all possible optical morphologies given a two-length scale extensibility. So we defined the set of possible, developmentally possible shapes given inflationary growth. However, a key question is, well, how does this compare to actual cells in nature? And so we had a collection of organisms spanning, the diversity of life, from the protists to the fungus to the plants. And we extracted their shape profiles and then plotted them onto this morphospace. And contrary, kind of surprisingly, the shapes of the naturally occurring cells do not populate the whole morphospace. They actually only stick to a certain part of the shape. And so this was extremely, interesting and curious, and got us wondering why. Why are growing cells in nature occupying the shapes rather than the other shapes?
Sorry, and in order to do this, you don't have independent measurements of gamma in each of the organisms. So you simulate fit to the.
Yeah, we fit just the shape.
For back where.
Yeah, because the experimental measurements are a bit laborious, they're laborious, and so we can just see, they fit this, how they fit here. Okay, so from the simulations, so the simulations are actually really powerful. We are able to extract certain quantities from the simulation. And one thing we can get from the simulations is a normalized radius of the shapes on this morphospace. And what we find is that the shapes that are preferentially occupied by naturally occurring cells tend to be thin relative to the shapes that are avoided. And we also got a normalized elongation rate out of the simulation. And what this normalized elongation rate tells us is that naturally occurring cells preferentially occupy shapes that are faster growing also. And so the way we interpret this is that cells in nature have been selected to have thin and fast growing shapes. And even more, if we try to understand this, what we think is happening is that this morphospace is representative of a fitness landscape, whereby extensibility profiles are under selection, and the readout for fitness is elongation rate, similar to the way growth rate is a readout for fitness in unicellular microbial organisms like bacteria. Okay, so but that's not the whole story. If you look at this radius flow, what you notice is something a little weird, whereby there's a rather abrupt transition from very thin shapes to really wide shapes. And this is very suspicious. So further analysis revealed that there is a instability, aka there's a bistable region here, whereby, or that corresponds to a cusp bifurcation in the morphospace, whereby for a certain range of parameter values, the extensibility profiles are able to generate two stable shapes, depending on the initial condition. And these shapes are on the permissive side where naturally occurring cells tend to be. There's a thin, fast-growing shape that is highly tapered. And then on the impermissive side, there is a wide, slow-growing cell shape that has this fallen knob. So it's like, it looks like it's bladed outwards and then started growing really slow. So that was really interesting. And if we look back at the species we had, there's a species called École bisexualis that has a shape similar to this one shape, which is, it has this highly tapered hyphae. And so a question we had was, given these predictions from the simulation that this shape is close to an instability, a mechanical instability, could we perturb the shapes of École bisexualis to, cross over tipping point? And the answer is yes. So we did some experiments where we broadly perturbed the shapes of the cells of École bisexualis using a Latranquilin B, which is an actin depolymerizer. And when we observed the morphology as a result, what we found is that we were able to find these wide, slow-growing shapes that are different from the thin, fast-growing control shapes. So this is a nice experimental validation of the simulation prediction. That was really satisfying data. Yes.
So in order to test this, it's beautiful. So in order to test the bistability, like that it's easier to go from one state to the other, you apply the perturbation test, right? And
yeah, I mean,
but how can you quantify in an experimental setup whether the stability is high or low? I mean, it has to be like comparative, right? So I would imagine that you would have to perturb the cell, the long in the thin and fast. You would have to perturb it with a small perturbation, and then you would have to take like another cell from more stable region, and then try to perturb it with the same strength and see that it doesn't affect it, right?
Okay, so you can perturb like, you know, quote unquote, the cells that have more stable shapes, which are the more rounded morphologies. With Lotranculin, they just get wider and more round. They don't become, and there's no prediction for how they shift.
So they can't switch states.
They don't, no. It's precisely the taper morphology that's predicted to switch state. That was predicted to behave in this way, theoretically. Yeah. However, so one last question from all this work is, you know, so why would actually bisexuals have these highly tapered shapes? So they're rare, first of all. Most species tend to have more rounded shapes. And so the question is, why does it have this? What's being selected for to make it go to kind of risk it all and be in sort of an unstable regime? And so we just have now an insight from the simulation, whereby if we quantify sort of the ratio of the radius of the simulation over its elongation rate, what we find is that there's a maxima of radius over elongation rate around the region of tapered morphology. And what this suggests is that this is a region that maximizes radius for a given elongation rate. What this tells us also is that tapered cells are also being selected to optimize for radius, or like the width of the cell, as well as their elongation rate. So it's not just that they need to be fast, they also want to be wide. So they are selected to be wide. And some intuition for this is that precisely the species that tend to have these tapered morphologies are mycelial hyphoorganisms like fungi and oomycetes that exhibit flow in their, they exhibit some kind of flow in their hyphae to transport nutrients or stuff to like neighboring or cells around them. So they're all connected in a network. And so based on fluid mechanics, what we understand is that wider vessels have higher flow rates than thinner vessels due to viscous drag associated with the wall. So potentially organisms that need to optimize, tip-growing organisms that need to optimize flow, cytoplasmic flow, also need to try to be wide. Okay, and hopefully, this is why there'll be a way we can test this in some way, and that'll be nice. But so far, this is a hypothesis. And with that, I just want to finish up this talk by just giving a few key takeaways. So remember, there's sort of a space of conceivable apical morphologies. There's all sorts of ways a cell could decide to grow apically. However, in this talk, we excluded certain shapes by defining a morphogenic process that's trigger-driven inflation. And we were able to define sort of all the possible range of morphologies given this morphogenic process. However, we're also able to see that by comparing to what's happened, where the shapes are observed in nature, that selection has acted on the space of possible apical morphologies to select for morphologies that are fast growing. And then on top of the selection for fast growth, we'll also see that there might be a selection for cell width for shapes that also maximize width at the same time. Okay, and that's it. Thank you. Yes.
Maybe while you take the last questions or next.
Yes, you should come in.
So super cool, thanks. So various organisms have different tip splitting.
Yeah.
I'm curious, your thoughts on that. Seems like this framework should be helpful for that as well.
That's actually a question I'm interested in, the way interested in. For one, so if you notice from the extensibility profile, I guess let me show it. For the pollen tip, they have a certain double maxima, like a local double maxima. And we hypothesize that they're able to tip split by separating, by increasing the width of the maxima between the extensibility profile.
Okay, let's keep going. Our next speaker is Wang, coming to us from Harvard, and you'll notice the large shift in topics, which will happen again throughout the day.
All right, thanks for the introduction, Bill. So it's very nice to be here. I'm John. I'm a PhD student in the technology lab at Harvard. Just make sure everyone can hear me, right? Okay, cool. Thanks. Nice. And today I'm very excited to share about our very recent work on using biophysical principles to predict fitness of SARS-CoV-2 variants. We're perhaps all just too familiar with COVID-19 right now, but however, we're all very less familiar with just how fast this SARS-CoV-2 evolves. So here I'm showing a graph. On the y-axis is the relative fitness or relative effectivity, and the x-axis is time. So you can see the Wuhan variant, the wild type, starts to emerge here at the beginning of the 2020s with a baseline of fitness equals to 1. And then just over a year and a half of time, the Omicron emerges, which has an effectivity 7 times actually larger than the Wuhan wild type. So it's much more infectious and millions of people infected. However, what was fortunate was that the COVID-19 pandemic was also the most well-documented pandemic over the entire course of human history. So sequences from all over the world are gathered and submitted to this very large database called the GICID database. And that allows researchers from all over the world to gain better insights into the pandemic. So now let's take a step back and just look at what makes the SARS-CoV-2 virus so infectious and what happens after the virus enters our body. So on the SARS-CoV-2 virus, there's a capsule. And on the viral capsule, there are things called spike protein. And in order for the virus to enter your cell and hijack your cell, the spike protein has to bind with a very specific cell receptor called the ACE2 receptor. And the very specific protein that is responsible for binding to the ACE2 receptor is called the receptor binding domain, the RBD, which is shown here. This is a crystal structure of RBD, and this is the ACE2 receptor, and this is their binding surface. So this RBD is relatively small, around 200 amino acids long, but it's very important for this cellular entry, and therefore it's also subject to the most immune response from human. So antibodies that your immune system makes will target the RBD, and once the RBD is bound by the antibodies, it cannot infect anymore and will eventually be neutralized by your immune system. So therefore, all of this just make the RBD a super crucial protein to investigate in the coronavirus scheme, and also all of the selection pressure just make this protein highly mutable. So now let's look at the experimental data. So there are 15 mutations on RBD between the wild-type Wuhan variant and the Omicron BA.1. And Michael Desai's lab showed that for Omicron BA.1, it has enhanced binding and escaping capabilities. And what they did is that they measured 2 to the 15 intermediaries between Wuhan and Omicron. So think about there are 15 mutations, and then each mutation could be either the wild type or be the mutant. So therefore, 2 to the 15th. And what they did is that they measure the binding affinities of all of those 2 to the 15 intermediaries to the ACE2 receptor, which is shown here, and to class 1 to class 4 antibodies. And on the x-axis here, I just want to mention is the minus log of the dissociation constant KD. So the further right you go, the tighter the binding is, and the lower your binding free energy is. And on this histogram here, the Omicron is shown in red, and then the Wuhan is shown in blue. We see that for ACE2, the Omicron has much tighter binding to the cell receptor, which is somewhat expected, but also most very importantly, it escapes 3 out of 4 antibodies that they measured. Yes? The different mutant, that's a good question. So that's a great question. So between the Omicron BA1 and Wuhan, all of those mutations happens on the surface of the RBD. So, and we'll talk that later, but those, since those mutations are on the surface of RBD, it doesn't affect the stability of the protein, so therefore shouldn't affect the structure of the protein that much. That's a great question, which I'll touch on. Yeah.
Is the RBD like more like a? Are there other evolutionary hotspots in the virus that are
besides RBD
that are as mutable as RBD?
Yeah. Actually, a lot of the spike protein itself is highly mutable, but the RBD itself, there are lots of mutation happens on RBD, but also on the other spots of the spike protein. Good question. Okay. Yeah. So now we have the binding affinities between the RBD with the cell receptors and antibodies. And the question that we ask is that, is there a physical model that we can derive that could capture the viral fitness or viral infectivity from those binding affinities that experiment from those experimental measurements? And to do that, we consider a really intuitive model. So first, starting from a unfolded protein, so all proteins needs to be folded in order to gain this function. So is the RBD. So first, the RBD has to fold properly in order for it to be activated and be able to bind to ACE2. And we denote that part of the free energy as the delta G fold. After the RBD is folded, it could bind to the ACE2 receptor and interest your cell. So we can denote that part of the free energy as the delta GA, delta G ACE2. Also, the RBD could bind to the antibody and be deactivated and neutralized. So we can denote that part of free energy as delta G AI. I indicates for different antibodies. And the mutant RBD might be more infectious if it has, first of all, fold tighter, or second of all, it has tighter binding to ACE2, just as we have seen from the experimental data, and also it might evade antibody bindings. So we write that using Boltzmann distribution, but so we have 4 states. So we have the unfolded protein, unfolded RBD, we have the folded free RBD, we have the RBD bound to ACE2, and then we have the RBD bound to the antibodies. And we hypothesize that the fitness of or infectivity of the virus is proportional to the probability that your virus is in the folded free state or in the state that you are folded and bound to ACE2. Because those are the ones who are eventually going to interest your cell, and those folded free RBDs are the ones that are being able to transmit through respiratory droplets. So if you write that out, on the numerator, you have these 2 terms, which correspond to folded and bound to ACE2 and the folded free. And on the denominator, you have the partition function, which is basically everything else. And in this very specific study, we can ignore the folding stability term, just as the great question that asked before, because all of the mutations that we consider are on the surface of the RBD, and therefore it doesn't affect the binding, the folding stability of the protein. But this is not always the case. So in other mutations might have, but just in this study, we can ignore it. And just by divided by the free energy of the folding term, we can get our final expression for the fitness or infectivity. And just wanted to know that in this equation, there are a couple of undetermined parameters. And first of all, C is the concentration of ACE2. So basically, how many ACE2 receptors are there in the cell? And also the Ki, which is the population concentration of antibody I. So let's think about this. If an antibody is very abundant in the population, then evading that specific antibody gives you a lot more infectivity, gives the virus a lot more advantage. And there is another scaling parameter A, and there is a hyperparameter temperature, which comes from the Boltzmann distribution. And to validate the model, we now look back into the data that we have. So we have, recall that Michael Desai's lab had those experimental data for all 2 to the 15 intermediaries between Wuhan and Omicron. However, not all of them exist, obviously. So we look at the overlap between experimental data and the GISAID population data, and those are, we can find the existing variants. And those are the variants that we both have experimental measurements of the binding affinity, and also we can derive the viral infectivity from those population data. So we took this data and then we split it into a test set and a training set, and we use the training set to determine these parameters. And we test this performance on the testing set, and we see overall we have a very high correlation. On the y-axis is the predictive fitness from our biophysical model. On the x-axis is actual fitness that derived from the population data. So this shows that a very simple or intuitive model using Boltzmann distribution who very well captures the biophysical relationship between the viral infectivity and binding affinities. Yeah. Yes, that's a great question. So basically you just plot out the growth curve of these, and you do just measure the growth rate. So for each variant, you plot the growth curve of how many counts you have over a period of time, and then you do regression on these and figure out the effectivity. No, this is coming from population data. So because sequences from all over are submitted to this mega database, so you can, in this database they are sequences and time and location. So therefore you can just aggregate those and coarse grain it, and then you can for each variant you can plot this growth curve.
And the variations in density of the coordinating and things like that are not so big as spoilers.
That's also a great point. The data is very biased, so that's actually a lot of efforts coming into clean up the data because, you know, for example, the US has a lot more testing than, for example, India has, but there is a very sophisticated way of doing this, which I'll talk more in the Q&A session, which there is another science paper that basically tries to, they use a lot of coarse graining method and basically trying to tackle this bias issue. But I guess the answer to your question is that the fitness are all derived from population data that coming from the growth curve. Yes.
Different question.
Yeah.
Which is like even in an individual, you're going to have many different antibodies.
Yes.
So how are you getting the data on the interaction? What do you mean by population concentration?
Yes. So it's a good question. So all of these data are coming from in vitro experiments from Michael Desai's lab, where they use yeast displace. So they displace the RBD on the yeast, and then they measure their binding affinity to ACE2 and all the class 1 to class 4 antibodies. And it is right that the human immune system is very complicated. But there are lots of different antibodies. But here we're only using these 4 antibodies because they first of all we have those data and they span across class 1 to class 4. Which is a diverse set of antibodies. Yeah. So what was your second question?
No, that was.
Oh, that was okay.
I just want to know what you're doing. Okay.
Okay.
Yeah. I mean, I think it's related. So it's shocking to me, like similar to my previous question, like that the binding affinity is clearly, like according to the graph you just showed in a couple slides. Can you go forward? Is so tightly correlated to fitness as measured from population level data. So what's the control for that? It's a correlation with a host of other mutations in the virus that is, I mean, presumably the entire virus is mutating, not just the RBD, like you said. So is the binding affinity a proxy for that evolution?
Yeah, that's also a great question. So this predictive fitness, this is infectivity here is not the actual infectivity. So is the infectivity contribution of the RBD. So this is not from the entire virus, but we can get the infectivity contribution from the RBD. Yeah, I think there's another question. Oh, okay. Yeah, but that's it. That's a great question. Yeah. I personally didn't do this experiment, but yeah, I didn't know. People might mutated spike protein and see how it be, but I don't think a systematic research exists on like the ablation studies of the spike protein, but yeah. Yeah, so we can further validate the model using the existing data, existing variants, and now we can train fit our fitness equation with the training setup without a certain mutation. In this case, G446S. And just a side note that G446S means it was glycine and became serine as the number 446 residue on the spike protein. And this will be the notation that we'll be constantly using throughout the presentation. And then we see we have a very high correlation, R squared equals to 0.92. And then we can do the same thing for all of the mutations that are the RBD between Omicron and Wuhan, and we consistently get a very high predictive power. Another very interesting thing we can do is we can do backtesting. So basically, we can fit the model using the data between the beginning of the pandemic to mid-2021, and then we can try to have the model predict the fitness of the variants coming after the threshold. And here we see that our prediction is fairly accurate, and especially we can also predict the jump caused by the Omicron BA1, which emerges at the end of the 2021. And you may ask, oh, but there's a gap here, and what causes the gap? And we looked into that as well. So in the backtesting, we find that our model predicts very well for the variants with a very specific mutation, T478K. However, it fails when that mutation is not exist in the variants. And we look further into this because this is a very interesting phenomena. And then we find that here I'm plotting fitness against binding energies minus log KD. And then we see that for ACE2 binding, there is indeed a fitness increase when we have tighter binding with ACE2. And by the way, these are all the sigmoid curves because of the Boltzmann distribution that we use. And however, for those antibodies, we didn't see a fitness advantage of those variants without T478K, especially at the early stage of the pandemic, escaping these two antibodies. So see here, your log 10 binding affinity decreases from 10 to like around 5. However, the fitness still stays at a plateau and doesn't increase. Yes.
So you trained your model on those 15 particular mutations from Wuhan to Omicron variant. Did you test it on like mutations that were not in those two, or was it? Yeah,
that's a good question. So we tried to do that, but the thing is that we didn't have more experimental data because the size measurement was all we had. But I think one thing that answers your question is this one out study that we did is basically we fit the model on things that without a certain mutation, trying to predict the effect with variants with that mutation. So this is like a proxy of what you prescribe, but a better way of doing this will be getting more experimental data and then trying to predict the infectivity of those. Yes.
This might be a silly question. So just so I understand correctly, so your fitness is just based on the growth rate of that mutation, yes?
So the x-axis, yes, the fitness derived from population data, yes, is based on the growth rate of the virus and regularized by some term in the basically that.
So maybe this is kind of silly, but also over time there's also the issue that human fitness against the virus obviously changed due to like vaccination.
Yeah.
Is that in any way factored into this model?
Yeah, that's a good question. Yes. I think particularly in this study, we didn't have the chance to investigate how the vaccination changes the fitness.
big variance comes in that graph and it have, does that have any, no,
No.
go back to the,
This one.
yeah, I mean.
Yeah.
Maybe I'm missing something, but so it seems like, does that variance at that point in time have anything to do with that or no?
This
Go back to the, it seems like error, maybe I'm misinterpreting this graph,
Yeah.
it seems like error jumped kind of significantly around 2021,
Uh,
right? Or no?
There's an error here, but this one's called by our mispredicting T47K. Yeah, so I think that's a field of question. So we do not know what happens during the vaccination events, but obviously, I think what happens is that it might be a coincidence, but actually we observed Omicron after the vaccination, so we saw that in this jump here. But this deviation from the actual fitness is caused by this very interesting mutation T47K, which our biophysical model failed to predict. And just going back to this T47K mutation is very interesting because it doesn't affect just in the measurement, it doesn't affect any binding affinity with ACE2 or with antibodies. And however, in the population data, we observed that variants with T47K has a huge fitness advantage comparing to those without it. And the effect of this specific mutation, I think, is still an open question in the viral evolution field. Another thing that we can get from our model is that we can evaluate the impact of Omicron mutant on viral fitness. And here I'm plotting the contribution of each mutation of fitness either towards evading specific antibodies or facilitating cellular entry, means binding to ACE2. And for example, here we see that N501Y, this specific mutation gains a lot of fitness by increasing binding affinity with ACE2. And for example, this mutation here, K417N, gains a lot of fitness by escaping this very specific antibody called CV6. And as a result, it gets punished by losing some binding affinity to ACE2. We can map all of those largest fitness contributions to the crystal structure of the RBD, and then we can for example, this K417N will be labeled orange here, and immediately we see that residues that are physically close to each other tends to have the same color, and that's just very reassuring that we see that emerges from our biophysical model. This also makes sense because those mutations which are on the binding surfaces between the virus and the RBD, where the ACE2 tends to affect those binding affinity the most. So that's why they typically clustered at on the structural crystal structures. The next thing I want to mention is that our biophysical model is epistatic. So what does epistatic mean? So epistasis means that the effect of one mutation is modified by the existence of one or several other mutations. And for example, let's consider two mutations, mutation 1, mutation 2, each have a fitness advantage of 0.1, 0.2. So in the linear model, the most naive model, if you have both mutations exist in your variant, then your combination effect will be 0.3. However, in the epistatic or nonlinear model, that's not the case. So these two mutations, although they are all beneficial individually, together they could be deleterious. And what we can do is that we can project our fitness function onto a like an Ising model or spin glass model, and then we can plot the interaction matrix here with epistatic coefficients here. So on the off diagonal are those CIJs, which is the epistatic term. On the diagonals are the CIs, which are the linear terms. So, and we can see here the same thing happens. For example, G339D and S371L are both beneficial if they occur on its own. However, together they are deleterious. Yes.
What besides we've measured is the effect on binding energies of the individual mutations.
Exactly.
So you're going to assume that the effect on binding energies is not epistatic, and then epistasis arises because fitness is a nonlinear combination?
Oh, both epistatic. That's a good question. So I think binding energy as a function of genotypes are definitely epistatic, but our model is also our model is a nonlinear map from the binding energy to the fitness landscape, which is also epistatic.
About the epistasis and binding.
Say again?
You don't know
Did
the
measure. I thought he measured all of
They measure all of it. Yes.
Yeah.
Yes.
You measured all
They alter all to the
Ah,
fitness. Yes.
very good.
Yes.
Okay.
Yes. And also a very interesting mutation is 1505H, which on its own is deleterious. However, combining with other mutations appears to be beneficial. And looking at this graph, you can also see that those with a very high Cij values tends to be or close to the tends to be close to the diagonal, and that's not a coincidence. So if you plot the absolute pairwise effect, basically the absolute value of Cij as a function of the actual distance, the Euclidean distance in the protein, you can find this graph. So we find that those that have very high epistatic effects are all those residues that are close together to each other in the RBD physically. Not necessarily on the sequence because the protein will fold, but in the actual Euclidean distance.
Go back one. I realize I'm super confused about this.
Okay.
Because the linear model would have said that I would add things, and that's certainly would be the first leading of it. And then I would have lots and lots of brown spots all over this plot.
No, the linear model will just be diagonal.
But is this correct? I mean, I guess I don't understand what's the off egg. Is that the total at that point, or that's only the contribution from epistasis?
This is the contribution of epistasis. Yeah. Another thing that highlights epistasis is that if we plot the fitness or effectivity as a function of number of mutations, so in the linear model, it would just be a linear growth as your number of mutations increase, and the biophysical model will tend to plateaus, which is a more physical model because your effectivity wouldn't go to infinite as your number of mutations increase. And all of these phenomena, I think, are very well observed in the field of viral evolution, but I think it's just also reassuring to see that these things emerges from our biophysical model. All right, and just to sum up, we developed a biophysical model that uses Boltzmann distribution to map the binding free energies of cells, receptors, and antibodies onto a fitness landscape. And we evaluate the impact of specific mutations on SARS-CoV-2 fitness, and the model is validated using experimental and population level data and may be able to generalize to other viral proteins. With that, I want to thank Professor Eugene Sheknovich for advising this work, and I also want to thank Maureen Hill, who is a wonderful summer student working on this project that hopefully is coming back to Boston next year, and Dr. Vaibhav Mohanty, who is also a collaborator on this project. I want to thank everyone in the Sheknovich group, and I also want to thank the funding agencies for supporting this work. Happy to take any questions remaining.
I have a question. Can I ask? Paul?
Yes.
So how much of the really great fit you get comes just from binding to A as opposed to the end?
That's a great question. Yes. So we did that as well. So that's basically ablation studies, whereas you just get rid of those. You just get rid of those terms, and then you see how well you fit. So we got like a R-squared around, I think, 26. Definitely less, because obviously you have less parameters, but also you are ignoring the escapes. And we find that in the ablation study that the class 4 antibodies contributes the most to the predicted power, which was the S09. Yeah, but that's a great question. That's Yes.
Why is temperature a hyperparameter? Body temperature?
No. Yes, that's a great question. Which Yes, that's a little bit tricky question to answer. And I have a slide here. So the temperature here is actually not temperature. It's actually if you write out the equation more carefully, it's actually effective temperature is the ratio between the temperature in the magnitude size experiment divided by something like our body temperature, something like that. If you write out the Boltzmann distribution more carefully. And so this temperature is very hard to determine, but what is reassuring is that we fit the model using a various range of effective temperature, or maybe should more call it effective energy scale instead of calling it a temperature. And we find that the temperature doesn't affect the curve that much, especially at the region where the actual variants are observed. So which is shown here, this is the fitness as a function of log H2. It's most of the existing variance cluster here, and actually it's more even so for the S09, where all of the existing variance clusters here. So we ultimately choose t equals to 1.6, such that when you are fitting with this parameter, the effective concentration are of the same order that observed in human serum. But yeah, that's basically how we determine the hyperparameter.
So our next speaker is Katherine Xiang, also coming from Harvard, and she's going to talk about geometry and biochemistry and cyclic domains. Please.
Thank you. Hi, everyone. I'm Katherine. I'm a grad student in Adam's Cohen lab at Harvard, and I'm going to be talking about cyclic AMP transport in cells. The picture here is the microscope that we took all of our data on. It's very pretty. And the image on the right is of the kind of data that we take. Specifically, we're able to map cyclic AMP spreading through culture neurons. And I'm going to put a video. You can see it spread out. So first of all, I'm going to discuss cyclic AMP in general. It's a small molecule secondary messenger in all mammalian cells, also in bacteria. And what I mean by that is that it's part of a very complicated signaling pathway. And most of the components I'm not going to talk about today. But our key player, cyclic AMP, which again is a small molecule, is circled here in red. And in general, I'm going to discuss three main ways that the pathway might be interesting or important. First of all, we know that all cells have to respond to environmental cues. Cells have to respond to cues in their environment, and what I mean is chemical cues. So when a chemical signal comes in from outside the cell and it binds to a receptor on that cell surface, information has to be transported or communicated from the outside to the inside. The way this happens is through a signaling cascade, and for cyclic AMP, which is associated to a lot of these receptors, it eventually gets produced. And one of the questions that we wanted to ask was just a very basic question. It's a small molecule, it gets produced. Okay, how far does it spread? And how fast does it spread? And we can also ask some more general questions about cell signaling pathways. For example, can these different receptors trigger different cellular responses, even though they all go through cyclic AMP? So one puzzle is if you have different receptors, but they all communicate through the same messenger, how do you get then different downstream effects? And these downstream effects can be things like cardiac cell-like contractility, regulation of heartbeat, increased metabolism, phosphorylation of various transcription factors, and so on. And on a smaller scale, we kind of also wanted to think about whether cyclic AMP's transport implies that, does it diffuse freely? Is it homogeneous through the cell? Or can even individual molecules of these receptors, so individual receptors on the surface, can those function as independent switches for signaling? I'm not going to get to all these questions, but basically the picture I want you to be thinking about is from these two extremes, is the concentration of cyclic AMP completely homogeneous, just spread out evenly throughout the cell when you get a response, or is it completely localized, where each receptor has its own little cloud of cyclic AMP and can then end up with its own little signaling pathway? The pathway is very complicated, and we're going to simplify it in almost a comical way because we're physicists. So we're going to think about, okay, how is it made and how is it degraded? And well, the answer is it's made from ATP through these adenyl cyclase enzymes, and it's degraded into AMP through these phosphodiesterase enzymes. So each of these enzymes can have a lot of different subtypes. So there are a lot of different kinds of ACs and different PDEs, and they can have different localizations in the cell as well. So they can be localized on the membrane or floating around in cytoplasm. And each of them is kind of the target of a lot of different drugs, because if you can control cyclic AMP, then you can control its downstream effects. So for example, PDEs are a very common drug target. Cyclic AMP also has its primary function, which is to bind and unbind to other proteins. The most important one is probably pKa, which is a protein kinase. So cyclic AMP can bind to it and activate it, and then that kinase can then perform modifications, specifically a phosphorylation on other proteins. But we're not going to talk about that very much today. So with this really simple reaction scheme, we can set up another simple reaction diffusion equation for cyclic AMP, where it diffuses in the cell and it's degraded by phosphodiesterases. And out of this equation, if you consider 1D, the simplest case, and production from a point source at steady state, well, we'll set the left-hand side to 0, and this eventually we get a decaying exponential, but one length scale comes out, which is known as a Thiel length, because Ernest Thiel cared about chemical reactors and like coined this term, but essentially it's a length scale set by the balance between diffusion and degradation. Okay, I'm going to discuss some of the experimental tools that we've developed to map and image, to produce and map cyclic AMP in cells. So we developed a tool for all optical production mapping of cyclic AMP, and we've termed it campsites, and it consists of two different proteins. One of them is an adenyl cyclase, which produces cyclic AMP, which is turned on by blue light. And the citation for that is here, but it was developed by other folks in 2011. And it's a protein from a soil bacterium that's naturally sensitive to blue light. So when you shine blue light on it, it produces cyclic AMP. And the other protein that we added to this genetic payload was pink flamindo, which is a red-shifted reporter of cyclic AMP. And it's a fluorescent protein which is attached to a cyclic AMP binding domain. And that means that when it binds to cyclic AMP, its fluorescence intensity increases. And in a certain range, that increase is linear to cyclic AMP concentration. So using blue, yellow, and yellow illumination and imaging and capturing light around 590 nanometers, we have a way to use light to do chemistry in cells. I'm going to show you an example of how this works. So we have our dish and our illumination. We use a digital micromirror device to pattern our light. So we have a little array of mirrors, and we can pattern the blue light. We use that to pattern the blue light in the way we want. So in this picture, we have 5 squares that are blue for half a second. And this is kind of just a proof of principle that we can do patterning, but we shine the blue light in this pattern and we image the cyclic AMP response, and the levels increase in only the cells in these regions. But this isn't a field of cells. So this isn't a dish with a bunch of hex cells. We want to be able to do this on the subcellular scale. So then I'm going to talk about how we did this and how we track cyclic AMP transport within a single cell. So first of all, we wanted to know how source localization can affect cyclic AMP transport, since we know that these sources are localized in different ways in the cell. And also, if we want to track diffusion, we don't want our thing to be making this, our thing that is making the cyclic AMP to be moving around, right? We want that source to be fixed. So then we made these 4 constructs where each pink flamindo or BPAC was localized to the membrane or soluble. So we have all 4 combinations. And then we did some imaging to make sure that indeed the membrane localized constructs were membrane localized and the soluble ones were not. Then we moved on to doing subcellular perturbation experiments. So this is a very similar sort of experiment where we directed the blue light to just this very small region of this MBCK cell. And these cells are long and skinny, which make doing these kind of diffusion measurements a lot simpler in terms of the modeling. So we turned on that blue light for just half a second and watched the diffusion through the cell. So in these regions 1, 2, 3, 4, the fluorescence goes as these traces, and these traces in each of these regions are fit to that reaction diffusion equation. So it's a solution in this kind of geometry to that reaction diffusion equation. And we found a diffusion coefficient that was pretty high, but what you would expect for a small molecule in cytosol of around 130 microns squared a second. We did the same experiment for cultured neurons as well. The data are a lot noisier because we're looking at these really thin noodles, but we found, again, a similar diffusion coefficient, kind of what we'd expect, 110 microns squared a second. Okay, yes. Yeah, so this data is from just this one cell. And it's a global fit to, so all these curves are the concentrations over time in these 4 regions. And it's a global fit for all the points to the same like diffusion coefficient and degradation rate.
And then on the right.
On the right, each point.
Different points of it.
Yes, each point is a single cell.
What's the error bar associated with estimating D in a single cell?
That's a good question. I think that error bar is, I don't remember specifically. I think
I guess what I'm asking is,
Tens,
do
yeah.
you think that the variations you're seeing are genuine cell-to-cell variations? Or do you think they're noisy?
I think they're most likely from measurement noise or from fitting noise. Yeah. I think it's possible that it could vary from cell to cell, but I don't expect that it would vary over like 2 or 3 fold. Yeah. Okay, so I'm going to discuss how cell geometry can determine transport and transport scaling. First of all, I'd like to consider some cases where we think about the localization of our source and our molecule that's doing the degradation. So if you activate the BPAC source is cyclic AMP, and that source is soluble, so the concentration is the same everywhere, it's just floating around the cytoplasm, and you have degradation from the membrane, then you'd expect the concentration in those like thin noodles to be lower at steady state than in that like in the soma of the cell. And that's because there's more, the surface area to volume ratio is like higher in those skinny noodles, and you're going to get more relative degradation. You can think about the reverse case where your production is from the membrane and your degradation is in the bulk. And in this case, because of the surface area to volume ratio, again, you're going to get concentration build up in those asperities. And I think this is a kind of not totally intuitive point, but a homogeneous stimulus can often cause heterogeneous cyclic AMP concentrations. And this is not just true for cyclic AMP, this is true for anything where you have production and degradation where one is membrane bound and one is soluble. Oh, what did I do? Okay, so geometry and enzyme localization can govern the spatial profiles of cyclic AMP. We then did some experiments to test this out. So this was also with a flash of blue light, so definitely not a steady state. But we were like, oh, we have the constructs, we can easily test this out. So we picked our membrane adenyl cyclase, and we gave it a homogeneous flash of blue light everywhere. And we tracked the concentrations in these different regions. And I don't want you to worry about the different shades of purple, but essentially the concentration in each of those dendrites was higher than in the soma for membrane bound production. We also tested this with soluble production. So we used the construct where the BPAC was soluble, and we did the same experiment on neurons again. And we found that the concentration in the soma was higher than the concentrations in the dendrites. And I think what this means is that actually we have both membrane bound and soluble PDEs, which we already knew. We know that these enzymes are localized to the membrane and in bulk, but we know that they're both active, and it turned out this way, which is kind of interesting. Okay. Next, I'm going to discuss this topic of surface area to volume and to diameter in more detail. So you can consider the wide field blue light stimulation at steady state. So we produce in some like, here we're going to model it as like a cylinder with some diameter d. And we have, now we don't care about the diffusion because we're doing it in a spatial, in a wide field way, but we have degradation and production. Kf is a rate that corresponds to production from ACs, and it implicitly, depends on the concentration of adenylyl cyclases. Kpde also depends on the concentration of degradation of phosphodiesterase. So our concentration is going to be proportional to the relative concentrations of each of these. And in in the membrane bound case versus the soluble case, those relative concentrations are going to be different. And we can figure out how that scales with the diameter of the dendrite in each of these cases. So if you're both membrane bound or if you're both soluble, there's no difference when you change the size of the tube. But if one of the partners is attached to the membrane, then you're going to get either 1/d or d scaling. So again, the enzyme localization and cell geometry will impact the concentrations of cyclic AMP. Okay, we can also look at if we have a point source that's activating a cyclic AMP. So instead, if you target the blue light to just a point on the tube, then we will care about diffusion, but we could also solve it at steady state. So if we set the left-hand side again to 0, then I already told you this, but we're going to have a decaying exponential in concentration with some length scale denoted phi. We can first look at the prefactor, and it turns out that the prefactor also cares about whether you're localized on the membrane or in bulk. But we can also look at the length scale, and the length scale also depends on if your molecule doing the degradation, if the PDEs are on the membrane or in the bulk. And the intuitive reason or the intuitive explanation for why this is the case is that if you have degradation on the membrane and you're in a thin tube, you're going to run into the walls of the tube more often than if you're in a thick tube and your degradation is on the membrane. So the teal length can also depend on the diameter of the tube. And for those electrophysiology nerds out there, this equation has the same form as the cable equation in electrophysiology, which is the distance, which has a length scale that corresponds to the distance that a stationary current is going to influence the membrane voltage in a neuron as well. And that scales as the square root of the diameter of your neurite. So we also tested this in our experiments because we wanted to know what that characteristic length scale of cyclic AMP is. So we did similar experiments where we targeted our stimulus to just that like one point on each neurite, and then we did some like quasi steady state measurements where we perturbed every 10 seconds, and then we waited for that distribution to go to what looked like steady state, and then we fitted an exponential along that dendrite. So you perturb, it spreads out, it gets to steady state. The video is actually paused, but we can take that steady state average delta f over f and fit a decaying exponential to the fluorescence and get a length scale from that, which turns out to be around 30 microns. But the more measurements we did, we found that it varies a lot. So it's not a constant, it changes a lot. And across different cells and measurements, it changes quite a lot with almost no correspondence to either that like d to the 0 or d to the 1/2 prediction. And we were thinking, oh, does it depend on which, like, on each individual cell? So this is the same data, but cells are connected with lines. The answer is not really. It varies a lot within a single cell. We also tested quite a few other parameters, so neural activity or whether there were spines. So spines contribute a lot of surface area, but not much volume. But we also found that these two didn't have any correspondence with that length scale. So we figured that it was most likely just very large fluctuations in like PDE, so enzyme localization that can cause like really large actually differences from almost like 5 microns to 50, 60 microns. So in conclusion, we developed a toolkit for performing all optical control of cyclic AMP. So we have BPAC and Pink Flamingo, which both generate and map cyclic AMP, and either can be membrane bound or soluble. And we mapped cyclic AMP transport in MDCK cells and neurons. We got these parameters, although there's a lot of variance on this length scale. And we found that it's geometrically compartmentalized in the cell. In fact, between dendrite and soma, for homogeneous stimulus, you can get variations of 2 to 3-fold. And the enzyme localization theoretically impacts concentration transport quite a lot through surface to volume effects. But we did not observe any like nanoscale domains, so like really small domains or distinct pools between membrane and cytoplasmic cyclic AMP. So what we think cyclic AMP looks like in the cell is that it's not completely homogeneous, but it's not completely localized either. It's something in between, and it's compartmentalized on this like length scale of the POA, around 30 microns, but can be less, can be more. And I want to say that these types of measurements can be done on other small molecules in cells as well, such as calcium or protons. I think those would also be very interesting. So thank you very much. I want to thank the organizers. Thanks for inviting me to speak. I also want to thank my lab, and I want to thank all of you for listening.
Yeah, it was an awesome measurement. So I guess this is probably kind of on your to-do list, but and I don't really know what cyclic AMP does in neurons, but you know, typically it's about global kind of responses to the environment. So the variability, you know, I also have heard that neurons are like not happy in tissue culture necessarily, you know, so yeah. I'm just wondering, like, can you do what does cyclic AMP do, and can you kind of, you know, enforce that and then do your measurements to kind of get a grasp on what the physiology,
yeah,
you know, that diffusion might play in these actual circumstances where the neurons want to respond to whatever they want to respond to?
Yeah, that's a good question.
Yeah.
So I think cyclic AMP's role in neurons is in synaptic plasticity. So if you have a lot of firing and calcium levels are activated, that's going to like change, I guess, the cyclic AMP profiles. But eventually, what I think happens is that like phosphorylation will recruit more more NMDA receptors to the like surface. So it recruits more receptors essentially. And there are folks in the lab who are trying to do experiments in slice. So and slice corresponds to like actual neurophysiology a lot better than culture neurons. But the report is very dim in slice. So there's some improvements. Yeah. There's some improvements too.
Great. I'm Katherine again. Great. So we promised these speakers from all over the world. So Mukherjee is coming to us from Paris, and he's going to talk about embryonic development. Please.
Thank you, Bill, for the introduction, and also for the organizers for putting up such a nice meeting. And can you hear me actually at the back? So as it's clear that the topics are quite diverging, and so maybe, you know, at times my presentation might become a little technical to my field, but I think I'll set up some main goals of this conversation today. So for example, what I will talk to you about today is what I'd like to call the topology and robustness in early fate decision. And there are basically three things I want you to take away from this, which are highlighted here, which is the role of topology and the biological concept of early fate decision. And maybe we can say something about robustness. Okay, so I think that's the thing. And then before I get into the main discussion for today, I want to tell you a little bit about the overall umbrella theme of my research, which is trying to understand the underlying physics of what I like to say form and flows. And it's essentially the question that if you start with cells and you put them together, eventually it gives rise to like a collective, which not only is, you know, just a collection of cells, but it also has certain function and it does something. And a big question, which is maybe physical, biological, but in some ways also philosophical, is when is the collection of cells become a functional entity, which is kind of interesting because it's also the center for physics of biological function. And I think most of my research over the years have been trying to understand how you build increasing morphological and functional complexity. So in simple terms, there are three questions that in some sort of hierarchy that I'm interested in: how cells make tissues, how tissues become functional, how these tissues kind of communicate. because if you do a function, you want to actually say that I have done these functions so that maybe other tissues don't do it. So over the years, I've been kind of working on changes of cell shape, interplay of fluid mechanics and cell biophysics, and actually changes, interactions of topology in different organogenesis systems where actually you build networks to be able to communicate. And the approach that I take to understand this sort of umbrella theme is that I borrow and actually develop ideas of geometry and topology, mechanics and flows, and some general concepts of active matter. So today, I think the topic that I wanted to talk about is related to functionality, but also it's deeply embedded in the idea of cellular identity. So for example, if you start with similar cells, at some point, if some of them become different, then you can do a combination of functions. And as you build different kind of cell types, combinatorically, the possibilities of function become really large, really fast. And we will kind of try to understand this question from basic principles. Now, of course, it's not a new question. It's a question that people are interested in, and they try to understand it from very different point of views. And I'll try to understand this from with the taste and actually interest, which I feel, let's say, most comfortable with, okay? So which is kind of a subset of problems where actually mechanics plays kind of like an important role in early phase decisions during development of an organism. And there are examples that exist, for example, in the context of oogenesis, where a collection of cells that actually makes the germarium of any animal, not all of them become oocytes, only some of them. Now, what sets actually this decision, whether you get to become oocyte or you wither and die? And there are also physical processes of sorting that are important in the context of developing the primary axis or division of cell fate in the mammalian embryo. And there is a peculiar kind of systems which in some ways are more primitive than are syncytial systems, where you have a huge number of nuclei that inhabit the same cytoplasm, okay? And but at the same time, they don't have physical boundaries. And in these systems, for example, since you do not have specific physical boundaries, like, for example, these cells do, how do you actually determine cell fate? For example, if it was some sort of chemical signaling, can you really localize it or not? So here, I think it gives rise to interesting physical questions to be asked. And I'll focus on actually this topic today. So the question is, if you have a collection of nuclei that can easily interact with each other, share the same cytoplasm, how does one become different from the other? So they started the same, right? What's happening? So to understand this question, we'll focus on, I'll give you a little bit of background in biology of this, which is a specific system, which is the Drosophila embryo, which is familiar to maybe some of you. And now the Drosophila is a model system for insect development. But if you really think about it, it's probably the strangest insect of all. And if you ask me later, I'll tell you why. So basically, it's a huge embryo that has one nuclei in the middle. And then it actually divides, okay? And in the beginning, it goes through a really rapid number of nuclear duplications. And then only at maybe like cell cycle 14, they reach the outer cortex of this outer boundary of, let's say, this embryo. And it actually makes cell boundaries. And then it becomes a proper, let's say, tissue or embryo, as you would like it. But what is left out in the bulk is there's some number of nuclei that people have over the years kind of ignored. And these are called the yolk nuclei. So in some ways, this is the primary physical division that actually happens first, that you get this yolk, which is here, and then the eventual embryo, which gives rise to the blastoderm, which goes through patterning and a subsequent morphogenesis. Now, as I pointed out, that there are synchronous divisions. And now if you ask yourself that by this logic that they're going through synchronous division, how many nuclei do I kind of expect at the surface? If the yolk nuclei was actually negligible, it would be 2 to the 13. Okay, but roughly what you see is actually significantly lower than that in the cycle 14. Now the question is, what is it actually about? And now, for example, biologically, what we can do, we can actually track these specific nuclei by advanced microscopy and tracking, etc., and track each of them and how they actually position from the bulk actually and get to the surface. Now, this is, I think, a process that can be studied. And then one important biological fact that I wanted to point out, which ties it to the nuclear fate, is to say, in the beginning of any embryo, there is no transcription. So you have translation because you have mRNA from the mother, you can actually make proteins, but you don't make genes, genetic expressions. Now at the cycle 14, where actually these nuclei get to the surface, the transcription kind of switches on. Now this is, and of course, if you wait long enough, we know that these yolk nuclei gives rise to various different genetic programs and different things. But then we started looking into this idea that there were publicly available single nuclei RNA-seq data. So you basically look at the gene expression at the onset of transcription, and you ask that whether their transcription is different. And what we find that even at the onset of transcription, the nuclei is actually already transcribing specific proteins that are completely different from the cohort that is actually at the embryo. Okay. So it means that the positioning that something stayed in the bulk and everything else ended up on the surface already kind of preposes them to give rise to different expression programs. So this is probably the first cell fate decision in the embryo. And it happens through a physical process of separating them in bulk versus surface. Now, this kind of boils down to this really complex question of fate and early fate decision to a question of physical separation, okay? So it kind of becomes a bit more tractable. So then the important question is what gives rise to the positioning of surface versus A? And of course, we can actually keep track of this nuclei moving in the surface, okay? In the whole embryo. And what we find is, let's say that along the process of this development, as they go through nuclear division, at some point between cycle 9 and cycle 10, you kind of get a bifurcation. So what we are looking at here is the distance from the outer boundary and each of these trajectories. Of course, there are some that are already happening here, but you can see a dramatic splitting in population. One actually moves more inward towards the bulk, and the other ones actually move outward towards the shell. Now, and the question is, let's say, what determines the eventual position of this nuclei and what gives rise to this bifurcation? Now, at this stage, there are no large-scale flows that are available actually in this cytoplasm. So the primary motion that arises is because of the fact that when a spindle actually splits two nuclei apart, the two daughter nuclei apart, they move like a force dipole, okay? And now imagine with respect to a surface, right? If you have a force dipole that actually separates, that is originally, let's say, tangential to a convex surface, what happens is both of the daughter nuclei, they move far apart, but they also move closer to the cortex or the boundary. But if you're actually tilted beyond a certain angle, what happens is one of them moves inwards, the other one moves closer. So that's one of the ways, that's probably the simplest way to describe this. And then, of course, we can calculate the orientation of these spindles, and which kind of gives rise to the motion that follows. And what we see, of course, is a distribution of angles with respect to the outer boundary that we can trace from the embryo. And of course, naturally, the simplest way to think about this, that what would be my orientation with respect to the cortex if it was completely, random. And what we find is that actually the orientation that by, let's say, randomness we would expect is less than 30%, that is actually orthogonal, but we find actually much less than that. So roughly 30%, but we find much less than that are actually orthogonal, which means there's a high number of tangential spindles that actually give rise to daughter nuclei that move closer to the cortex. So here actually then the question becomes that what sets the spindle orientation rather than the motion of the nuclei. So now spindle orientation in a cell biology point of view has been studied over the years in many ways. Now, mostly in the context of a cell, because the cell has a boundary and the spindle actually has asters and these mechanical elements that are built of microtubules, and it interacts with the cell boundary. And that gives it some sort of geometric cue of how to position itself, okay? And in many ways read spatial constraints, etc. However, here, the system is very different because you don't, before you reach the boundary, you don't really have spatial cues from the boundary. The only thing that you have is actually other spindles and asters. And what may give rise to is actually the interactions between them. And of course, I show you kind of for illustration these beautiful pictures from, I think it's the starfish embryo. And there are these huge asters that make these interaction zones of how microtubules actually overlap with each other. And microtubules are actually this, let's for the simplest way, stiff polymer. And when they interact with each other, if they're polarized, So if they have anti-parallel overlap, there's a molecular motor that actually goes there and tries to slide it outward, kind of can generate a force. Okay, so this is a very simple concept, and you can kind of think of it as, let's say, two, let's say, nuclei with esters, and when they have overlap zones which are anti-parallel, then you need to kind of generate a force to kind of push each other apart. So with this idea, we can actually try to understand how any of them move at all to begin with. Okay, so the simplest question. So this can be captured with this idea of actually making overlap zones and actually the motor generating force. And essentially what it gives rise to is a repulsion. And this repulsion, of course, you can, kind of trace it back to some sort of a relaxation in a potential problem. And of course, because of the curvature, it gives rise to an outward force, and that actually inflates the balloon. And of course, you can kind of simulate it and study this. Now, one of the interesting things that actually I want to point out here is, let's say, that in any of these such, let's say, inflating structure, if you start with spherical and you have some sort of bending rigidity, etc., one of the shape changes that you see is actually that it becomes more elongated, more prolate in some ways. And I can discuss why in some cases and what are the conditions that you need for that. But now, what we actually see in the embryo is a bit different. And it's to do with the boundary. For example, here, this blue thing that you're looking at is the original, let's say, envelope of this nuclei in the beginning, let's say an initial condition. And then it actually reaches something that is the embryo shape. Now, in this case, for example, the ellipticity actually decreases. And it is a lot to do with the fact that actually in the beginning, let's say these two poles are already too close to the boundary, and you start interacting with them. Now, if the mechanism of this ester repulsion is kind of true, if I change the initial conditions where the subpopulation doesn't interact with the boundary, what I would expect is actually an increase in eccentricity before it actually reaches the boundary. And what we find, which we can do actually, that if we change the initial conditions, it actually increases in eccentricity or the ellipticity of this ellipsoid, let's say. And so this kind of is some sort of basic principle proof, and it's actually not a fit. So basically, there is only one time scale. So I remove the boundary, which is in the normal wild type case, and I fit the wild type with one time scale. And then I predict what it would be for the other case. Yes.
I didn't really understand what causes the repulsion of it.
Yeah, so essentially think of it this way. So each nuclei has two like emanating esters. And whenever neighboring esters, overlap with each other, so they're polarized like my fingers are. There is a tip and there is a stop. No. So basically, when you make a physical overlap, two polymers, there are molecular motors that can go there, and then they slide it apart. So that's the force generation, and essentially it's a repulsion with a very short length scale. Essentially, so the moment you fall off, you don't feel each other anymore. But on the other hand, if you wait, if the cell cycle is long, these polymers also grow. So it's a dynamic problem. You can solve it that way. Yeah, sure. Yes. Yes. That's one length scale. The other one is how large are these esters? Yes, of course. Yes, I'll come to that. I'll roughly answer that question. So basically, this kind of also alludes to the possibility, as our audience pointed out, that perhaps there is the density effect that at high densities, when pushed together too much in a constrained space, for example, in a sphere, you might get instabilities that actually gives rise to a flip. And it is a familiar phenomenon, which, for example, exactly what I tell you. It's a familiar phenomenon, which is some sort of localized buckling. Now, to understand that, what we need to do is kind of look into the data and actually map out the geometric aspects of it. And when we kind of do that, we can map out all the geometric aspects like density. And for example, we can ask that whether there are regions which has more localized strain compared to others, and try to relate it with the spindle orientation. And okay, so just simple things. But the basic idea is, let's say that one interesting thing to notice is, let's say that although the spindles are oriented objects like rods are, you don't really see any sort of nematic order, which at high densities you would expect. Okay, so that probably says that maybe the density is not high enough. Okay, that's one aspect. The other thing is that if that argument was true, that because of compression you get localized buckling, when I try to relate the tilt angle of this spindle with respect to the local strain, I would expect a cloud here, which I don't. Okay, so basically I see a more homogeneous distribution. Now this gives rise to the alternative possibility, which is that as these overlap regions interact, maybe the other possibility is that they also align. So and this is intuitive. So I'm gonna try to kind of do this. So imagine you have with my fingers, okay? So you have this thing, and in principle, there is no asymmetry between top and down. And I try to slide it apart, okay? So nothing happens. Now, if I'm slightly tilted, with respect to the other one, one of the things that happens is, let's say you have an unbalanced force towards inward between the top and the bottom, which kind of does this, okay? Now, if I keep twisting too much, at some point I'm making overlaps that are as such, and then I actually give rise to like a barrier crossing kind of instability. So that's the basic idea. For this one, what needs is, so basically, you can consider a 3A setup. And basically, what at a single microtubule that you would expect is, let's say that you have these overlaps and you can have an alignment. Now, this is something that is known for simpler microtubule structures, for example, the yeast microtubule structure, and you can actually see this alignment. Funnily enough, it was actually my wife's PhD thesis, which I didn't know for a long time. It was a sad car ride home, actually, but anyway. So now the question is, as at the microtubule level, you can actually give rise to such alignment interactions. In this complex array of structures, how do you actually think about the geometry of this? So that's what I think there. Yes.
Just a quick point of clarification.
Yeah.
So this is assuming equilibrium. Is that right?
No,
I mean,
no.
Oh, it's dynamic.
Dynamic.
Okay.
No.
Then
So
it seems like then all bets are off for with respect to this buckling and everything. I mean, buckling could happen, but in a non-equilibrium system.
No, so I mean, for buckling, you need to develop the strain enough so that you can actually get a buckling. The main thing that it says is, let's say you're at a regime where the density is not high enough to constrain the strain, and which kind of makes sense because you're pushing each other apart. You're free to kind of expand. You don't localize strain as much because you can move to kind of relax it. So that's what it means. Now, if I take this same system and artificially put it in a, let's say, more elastic or more, things where the time scales are actually slower, then you can localize it because you don't relax the strain. So that, yeah, perhaps that's what it alludes to. But anyway, so basically what I wanted to talk about is, let's say that we have this possibility of this spindle alignment, which I point out, and of course you can map out in terms of the stability of these structures. And as I pointed out, the intuition of it that, of course, at some point, if the tilt is too much, you kind of flip, okay? So it's like a barrier crossing phenomena. This is a kind of this nice mechanism where, an aligned object, if due to fluctuations, if it creates small tilt with respect to each other, and you can actually align back, okay? Now the question is, this kind of leads to another problem, which is, I need these overlaps to be able to align. At the same time, I'm pushing each other apart. Now, so as a result, the distance is increasing. Now, do I have enough time to actually give rise to a proper alignment? That's one. And the second is typically these sort of interactions would decay with some sort of length, okay? Now, how can you still align as the object inflates and the distances increase, okay? So that's a question that I need to actually solve. And here I have some simple propositions. So basically, the total alignment or this torque depends on how I integrate all of the forces along this shell with all of my neighbors, essentially. And this is the basic idea. And for that, what we need to consider is, let's say, torque generation at each of these microtubule overlaps, which goes as, let's say, the length of these long microtubule fibers, let's say. And the other thing is, how many such overlaps do you make? And for that, I make use of a very simple argument, which is that these are emanating asters. And I would assume that it drops off as 1/r², okay? Just from centrosome along, Kepler's Gauss's law, whatever you want to call it. Now, each torque element, which I kind of describe here at the boundary, then is related to some basic force, some width of the shell, and the number of emanating microtubules, and how the density falls off. So because density of overlaps will depend on that. Then times the length scales of this each microtubule, and then the arc length here. Okay. Now this thing has an interesting property that if I rescale this entire system with some sort of, let's say, factor lambda, you will see that it actually falls off. Okay, it doesn't care. Okay, it's primarily because now the other thing that it says, so it says that the alignment with respect to on the cell, with respect to all integrated torque with respect to all neighbors, does not really depend on, at the lowest order level, does not depend on the distance, only some sort of geometry. The other thing that we know is, let's say that this ds over r, it's essentially, and I integrate over this entire arc length, it gives me an integral of essentially the curvature of this boundary. Okay. In 2D, this is related to the Gauss-Bonnet theorem on 2D. And now you can try to do the argument in different ways, whether you want to force balance or not. But the basic idea is, let's say that the integral of this thing is related to the Euler characteristics of this polygon, plus the defect that you make with respect at each corners. So essentially, curvature is how fast my tangent orients, okay, moves. Now each boundary jumps. So you need to take into account the defects that you get actually each of this boundary. And if you, so independent of how exactly you actually do this, in all cases, what you find is that the torque, this integral is proportional to the number of neighbors. Now the prefactor changes with respect to how you make the argument and also the intercept. And then of course you can actually do a dynamic, like a simulation where you actually have polar objects and you actually do stochastically this, and you can actually map out the residual torque with respect to the topological charge. I realized that I need to, so this is one of the main results and I need to speed up, okay? So now of course this suggests that there is some sort of connection between the spindle tilt and the topology. So what we check, of course we can do that because of the mapping out the geometry, that perpendicular spindles are more negatively charged, okay? So we can actually do that. Negatively charged means, let's say less than 6 number of neighbors. Positively charged means more than 6 number of neighbors. And then the opposite is also true, which means that negatively charged regions are more perpendicular. And this is an interesting thing, that if I ask that based on the topological charge, what is my probability that I'm going to be tilted more than 45 degrees? I see, for example, a decay with respect to negative versus positive, which means that the more number of neighbors that I have, more alignment, and I'm actually more tangential. Okay, so that's a simple deduction. And then, of course, this alludes to something. So which means that, in this system, where shape and size is kind of fluctuating and things are expanding, you need to kind of add up the morphogenesis or the changes into this shape and size in this very directed way to change actually the topology, which is a harder thing to change. And this has something to do with, let's say, robustness in other cases, and it has been alluded in, for example, flocks of birds, that topological interactions are more robust than metric ones, which kind of makes sense. Now here we have this system that actually kind of does it naturally, which actually sets its topology, and there are constraints. And one of the predictions would be that, so of course we kind of find a relationship between the topology and the tilt. Now what we need to establish is whether it actually does not care about the scale or the length scale or not. One way to do this is to slow down things. So this is a very fast process. So how long actually I make this connection and how long these fibers are depends on the length of the cell cycle. That's the limiting factor. So ideally, we can actually do that. And of course, we can do that with genetic manipulations by cell cycle genes. And what it allows you to do is actually play with the amount of inflation and the amount of actually how much this each domain or what is the length scale between, let's say, different nuclei. So you change the density. And you can dramatically do that, actually like 2 folds or something. And what I will propose here is, let's say that here you can collapse with all of these variation in actually densities into one master curve. This line is actually a fit to the original wild type data, and everything else actually just relies on it. So this kind of establishes that this alignment at this surface level does not depend on size or some sort of length scale, but at least on the lowest order and local topology. And I think with that, I'm almost at the end of my talk. And I wanted to kind of give an overview that, we have a nice way actually to understand how the shell forms and inflates. And there is a relationship between the topology and scale independence with the tilt, and how that actually gives rise to some sort of regulation of fate. And with that, I'm at the end, and I want to thank my experimental collaborators in Duke and my theoretical collaborators in Maxim, Nicolas Supérieure, and Sebastian Streichen and Noah at UC Santa Barbara. Thank you.
Good. So we told you that people come from all over the world. You may not realize that getting people from different institutions in New York City to meet each other is actually always quite difficult. So it's a pleasure to have Danil here from Columbia to talk to us again about those.
But yeah, thank you for the intro. Thank you for the invitation. Thank you for organizing this. So I'm going to talk to you about two memory processes that occur. One of them is memorization, seeing things in the world and storing them brute force, and consolidation, seeing many examples of things and trying to create a generalized representation of them. The model that I will use to do this is associative memory networks that Tom just talked about. This was work done with Kim Stackenfeld from DeepMind, Dima Krotov from IBM, and my advisor Larry Abbott from Columbia. So the general setup is the following. As you walk around in the world, you see gazillions examples of one class of regular items, a common class, and you occasionally might see one example of an item from a rare class that I'm going to call an exception. And the idea is you're not going to be able to, or you're not going to want to verbatim store every single item from the common class. There are simply too many. In contrast, when you see one of these rare items, assuming that this is something that is interesting enough for you to store, so I'm going to take the assumption that you do want to remember everything in some sort of way, you're going to have to store this verbatim. So the way that I operationalize this scenario is the following. I'm going to have a data set of R plus E items. Capital R of them are going to be regular items. Capital E are going to be exceptions. R is going to be much, much larger than E. So, and this data set is going to be much, much larger than the memory capacity of the neural network model that I'll tell you about. So you're going to need to somehow compress this data set. One option is to throw away all the exceptions and just do what's called statistical learning on the regulars. However, the premise is you do want to store this. This is somehow interesting. This is somehow important. So what we're going to try to do is we're going to try to get our model to consolidate or compress the regular items into some featureized representation, and then we're going to try to get it to store the exceptions verbatim. Now, this type of scenario typically breaks standard machine learning models. If you do sort of any vanilla like neural network training 101, what that neural network is going to do is it's going to completely ignore this. It's just going to attempt to do statistical learning on the regulars. So we're going to attempt to remedy this. So the actual data set that I'm going to use, this is the MNIST digits data set. So I'm going to use 6,000 zeros. I don't know if you can really tell, but each one of these is a handwritten digit 0. And this is going to be the regular class, and I'm going to have 3 MNIST ones that count as the exceptions. Okay, so this is the data set. This might be a good time to pause and ask if there's any questions for sort of like the premise of what I'm trying to do here. Yes.
Have we made any assumptions about like the number of like rare classes compared to regular classes?
So right now there are only 2 classes. One of them is regular, and there's a gazillion examples. There's 6,000 examples, and one of them is the exceptional class. That is the a gazillion examples of regulars, and then 3 exceptions. So there, yeah, only 2. Yes.
For the exceptional class, do you have like pre-specific
Both. You can go either way. Right now, like what's going to turn out to happen is they're all going to be distinct items. But yeah, there could be like, because I'm using 3 instead of 1. Like there could be one exception, and then you can't do any sort of consolidation on those. You can do a little bit of consolidation. It's going to turn out that you're going to just memorize these, all 3 of them perfectly. Yeah, good. Yeah.
And what does success mean? What's the thing you asked your network to do?
I'm asking the network to retrieve the item, and the way that I'm going to ask it to retrieve the item, I'll talk about this in detail later. But the same way that Tom does it, present a perturbed version of it, and then get it to complete the full version.
Maybe I missed it. The regular class is 0.
The regular class is 0. Yeah.
Okay.
By the way, if you train a network with zeros and you give it half of a 1, what does it do?
It'll output a 0. It'll output the closest 0. Or it'll output some metastable state. That's weird. Cool. Good. Thank you. So yeah, so let's talk about the actual network that I'm going to use to attempt to store these. So the network is the so-called modern Hopfield network. This formalism was proposed by Dima Krotov and John Hopfield pretty recently. This is a generalization of the dense associative memory. It's generalization because you can choose the nonlinearities here and here to reduce it to either the dense associative memory, the classical Hopfield network, the modern Hopfield network as coined by other authors, Ramsauer et al. But the structure of this network is as follows. So it is a recurrently connected neural networks. There are two layers of neurons. So I'm abstracting away the specific neurons, but there are n neurons in this layer. N is equal to the number of pixels in an image. M is a freely chosen parameter. M, I have 100 here because I want the capacity of the network to be much smaller than the data set size. The 2 layers are the visible layer V, the hidden layer H. The activity in the visible layer V is projected through this synaptic weight matrix W to the hidden layer. This represented by H. This H is the input current into the hidden layer, which is then transformed into a firing rate F through this nonlinearity, which I'll talk about a little bit in a second. Then this firing rate is then recurrently projected back through the transpose of the same weight matrix back to the visible layer, which is then this input current V is then transformed into a firing rate G through this nonlinearity. So this nonlinearity is a pretty simple one. It's just spherical normalization. You don't really have to have it. It's nice for the training procedure that I'm gonna talk about. This nonlinearity up here, f of h, is kind of interesting. So if you're into machine learning, this is the same function that's used in as the attention mechanism in transformers. But the softmax function is, Tom mentioned this, I think John also mentioned this. This is exactly the Boltzmann distribution. I don't use it as a Boltzmann distribution, but if this is the equation for the softmax. The thing that I want to point at here is this beta parameter, the inverse temperature, which allows us to tune the specificity of this hidden layer representation. So if beta approaches infinity, this softmax approaches a literal max. So what's going to happen is the single element of this vector that its maximum is going to be 1, and all the others are 0. As beta goes to 0, this vector is going to be exactly uniform. So it's going to be 1 over m for all the entries.
Why do you need to have like W and just for not just like W and another matrix?
Very good question. The symmetry allows us to write down an energy function. So if there is a symmetry, then you can guarantee that these are going to converge to stable fixed points. If there is slight asymmetry, empirically, it happens to be that it also does converge to fixed points. If there is extreme asymmetry, it might not. It might oscillate. It might do chaos, and there's no, you don't know what's going to happen. But yeah, Yeah, that's as far as biology goes, that's a kind of a buck wild assumption that there's just like perfect symmetry, but it allows us to do math on this, and however this math I won't tell you about, but that's why it's symmetric. Yeah, good question. Cool. Any other questions about sort of the structure of this network? Nice. Okay, so how do you drive this thing? And the way that we're going to operate this is similar to what Tom described. So I'm going to take as an input a perturbed image. So I'm going to randomly take half the pixels in one of my inputs, set them to 0, and then I'm going to initialize at time 0 this visible layer to be that perturbed image. And I'm going to run these dynamics recurrently until they converge to a fixed point, which is now guaranteed by the symmetry. And then that fixed point, G star, is now going to be the output. What we would like the output to be is exactly the original unperturbed image that we perturbed to create this input. So how do we get this network to achieve this target? And so we're going to train this using sort of machine learning neural networks 101 techniques with stochastic gradient descent. The way that we're going to do this is we're going to grab a batch of items, so some small random subset of the data set. We're going to define a loss on that batch. So we're just going to take the sum over individual losses per item. So this is the target, this is the output. Where, for example, this loss might be the mean squared error between these two vectors. We're simply going to sum them up, and then we're going to update the parameters w to minimize this loss. So we're going to update them proportionally to in proportion to the negative of the gradient of this loss with respect to the weights, in an attempt to minimize the loss over the entire data set. So that was machine learning 101. What we're as I'll show in a second, doesn't really work. So we're going to go to machine learning 102. And what we're going to do is we're going to take this loss and we're going to do something kind of potentially interesting to this. We're going to reweight the loss. So different components of this loss are now going to contribute differently, and these alphas are going to be the scaling factors for how much each of these items is weighted in the loss. And assuming we know a priori which classes are rare and which classes are exceptional, we can set these weighting factors, alphas, inversely proportional to the frequency of occurrence. So if it's a regular item, this R is going to be large, so this weight is going to be small. If it's an exception, this E is going to be small, so this alpha is going to be large. So we're basically going to be giving a lot of importance to exceptional items in this loss, and we're going to be downweighting the importance of rare items. This is effectively what using this ground truth. What this is effectively doing is it's taking the average loss on all the regulars, the average loss on all the exceptions, taking the average of those two. Because the premise is the exceptions are as important as the regulars to store in memory. Another technique to sort of deal with this problem from machine learning 102 is to resample the batches. So right now, what we have been doing is we've been assembling batches by randomly taking a subset from the data set and then doing that over and over and over again to update the loss iteratively. Now what we're going to do is we're going to still randomly but non-uniformly assemble batches. So we're going to take the probability of putting an item into a batch. It's going to be, again, proportional to the inverse frequency of the item appearing in the data set. These are kind of standard techniques in machine learning. This is a nice review of them. The question is, can we use them in associative memory networks? And the answer is yes. So if we do loss reweighting here, This is now going to be the loss over the course of training, because we're iteratively stepping through this gradient descent to update the W parameters to minimize the loss. You can see in here in solid, the solid lines are the loss over training for using loss reweighting, on the red being the exceptions, the blue being the regulars. So the loss on the exceptions goes to 0, which means those items are perfectly stored. The loss on the regulars goes to some small numbers. They cannot be perfectly stored because there are so many of them. So there's going to be some error. However, the error is pretty low. Now, if you do the naive version where we don't do any loss reweighting, then the regular items are still learned pretty well. The exceptions, however, are a disaster. So the error on the exceptions is very, very high, even at the end of training. And if you keep training this forever, it'll just stay flat. So this kind of says that, machine learning 102 is necessary to get this problem to work. And you can also do this with importance sampling. Now, instead of reweighting, we're resampling, and you get kind of the same pictures. So now I want to get to the crux of this, which is in real life, when we walk around and we look at stuff, we don't know a priori whether this item is a regular item or an exception. We need to somehow infer this. We don't have the ground truth. The question is, how do we get those alphas, those weighting factors, or those sampling probabilities without prior knowledge? Here is one set of intuitions. If it is a rare item that we're attempting to memorize verbatim, what's going to happen in the hidden layer is there's going to be a very localized representation, meaning there's only going to be a few, or in the extreme case, one hidden neuron that is active when retrieving that memory. And the reason for that is when this hidden neuron is active, only these weights contribute to the output, so we can basically store whatever image we want in these synaptic weights verbatim. And then once the neuron is active, that image is going to be then projected into this output layer. What this means is in the hidden layer, there's going to be a low entropy. This is going to be a essentially 0, 0, 0, 1, 0, 0, 0, 0 distribution. It's going to be a delta function. That means the entropy is going to be very, very low. And for low entropy, if we want to memorize, as I mentioned already, we would like to set a high learning rate, a high alpha. Conversely, if it's a common item, that means we're going to have a distributed representation. So we're going to have several hidden units contributing. So we're going to sum up whatever values are stored in these synaptic weights to produce the total output. If there are many hidden units active in the hidden layer, that means the entropy of that hidden layer is going to be high. So we want a low learning rate. So to summarize, the alphas should be proportional to the negative entropy of the hidden layer for any given item. And it's proportional. There's some normalization that I won't tell you about. It's not hugely important for this premise. So that was just the intuition. The question is now, does it work? So let's see if it works, just assuming that we know the ground truth. Like, is this intuition even valid? So we're going to train the network using the ground truth information that I told you about a couple of slides ago. And we're going to measure the entropy over the course of training of the hidden layer for exceptional items and for the regular. And then, as you can see, the intuition kind of holds up. Over as we learn exceptional items, the entropy in the hidden layer representation of those items grows low. The entropy for the regular items remains high. Sorry. Yes. Has 100 units in it, yeah. It is a one-dimensional distribution. It's a length 100 vector, and that is the distribution because it is in fact the Boltzmann distribution. So it's a well-defined probability distribution. Entropy. So we've trained using the ground truth, and the entropy does separate. Now the question is, can we bootstrap this? Like, it's not obvious that if we just stuff entropy into the alphas that it's going to work in the way that we expect. But fortunately, it in fact does. If we set the alphas proportion to the negative entropy, we can see that the entropy of the exceptions goes down. The entropy of the regulars stays high. So this looks to be like a good signal for switching from memorization mode to consolidation mode, or like statistical learning / compression mode. Does it work? So I'm going to again now show you over the course of training. Now I'm going to show you the evaluation loss. This is no longer the loss that I'm training on. This is the ground truth loss. However, I'm training using entropy. And the exceptional items get memorized. The regular items get consolidated. If we're using loss reweighting, if we're doing importance sampling, same thing. It might take a little bit longer here, but overall, it does work. So that's kind of the crux, is finding signals that are built into the network to attempt to both memorize and consolidate. Question.
Do you know when you get kind of results decreases?
No. This is just I would sort of chalk this up to like machine learning, gradient descent, loss landscape quirks. Like the loss, like it's very hard to visualize this loss landscape, because you have like 100 times 784 independent variables that you're moving around, and you're trying to minimize the loss over this. So there might be like weird cliffs in the loss landscape that you take like one step of gradient descent and suddenly your loss is way better. But I wouldn't sort of put a lot of weight on this, because if I run this optimization again, the cliff might be somewhere else. Yeah, good question though. Okay. Any other questions? So that was kind of the setting up the premise. I'm going to do sort of more of the same.
Can you repeat why
Yes.
it is intuitive that for items in the regular class, the activity in the hidden variables is spread?
Yeah. Good. The intuition is, because we are unable to store a single item verbatim, so we can't just like activate this neuron and then take the outputs of these synaptic weights and drive the visible layer with it. We need to sort of build features that we can combine in different ways.
Okay.
So if we have a single item, we might have like this neuron corresponding to exception 1 and this neuron corresponding to exception 2, right? But then if you have 100 regular items, you'd need to like do like a combinatorial kind of code up here to represent those common items.
Memorize like one single item. The best way is to just like say, if this neuron is firing, that means like that's the one that I saw onwards.
Like because that item is perfectly stored in these ways, whereas like you don't need to do any sort of like combination. That is the intuition. It doesn't actually have to be this way. You can actually store both of them in a distributed way, but it empirically turns out to actually be this way. And this might depend on the data set. This might depend on how it's trained, but for this data set, for this training paradigm, it seems to be that way. Nice. Yeah. Any other questions? Yes.
Trying to make sure I understand this correctly. How robust would this model be to like 2 classes of common items? Like, would it be able to capture?
If there are 2 classes of common items, you don't actually need to worry about it. If you want to do 2 classes of common items, then you do machine learning 101. You do this, and then because both of them are sort of equally balanced in the data set, those are going to be by default equally important. So if you throw away half of them, you're going to get major hit to the loss. So you need to incorporate both of them. So it's going to have to sort of incorporate both of them. yeah, the crux is that like one of them is really, really small, but it's still important. And we don't know a priori which ones are belong to the really, really small house. Yes.
What do you think the limit is? He says like 50-50,
Yeah.
Is there like percentage that it gets too obvious?
I don't know. I think most of what people talk about is like these two extreme cases of like where it's like balanced, and yeah, 49-51 probably still solid. 25-75, I don't know. 99-1, like you're in trouble. So that's yeah. Sorry, that's not more informative. There's one question. Yeah.
This might be a sort of a bad question, but so it sounds like the motivation here is basically this looking forward to biological instantiation of this, that you need to be able to sort, is this a common item that I want to compress or keep it as an exception? But clearly there are examples of classes of things like human faces where you're seeing a lot of them, but distinctive power is extremely important. And you don't want to actually compress, even though you're seeing probably dozens of them every day that are new. So I guess, yeah.
Yeah, that's super good question. Yeah, so the question is like, more generally is like, what do we count as exceptions? And right now I'm calling exceptions like things that are wildly different. It could be possible to consider exceptions to be like particularly important instances of cats. Like you want to compress cats, but you want to memorize Garfield the cat and Grumpy cat. And yeah, how do you tell those apart? That's harder. I don't know. But yeah, this is a thought that I've had. The answer is TBD. Stay tuned. Good. Okay. Onwards. Okay. More of the same. So, okay, we've talked about alphas. If we use the alphas to be the ground truth, everything's great. If we use the entropy of the hidden, negative entropy of the hidden layer, we're still good. Let's try some other stuff. What if we set the alphas actually just equal to the loss parameter? So this is effectively just taking the loss and putting a square in the loss. What if we look at the number of time steps that it takes to converge to the fixed point? What if we look at, instead of the entropy of the hidden layer, we look at the max of the hidden layer or even the norm of the hidden layer? And the answer is these all kind of work. So this is the entropy, assuming that we're training with ground truth. This is the entropy, which you've already seen. And now assuming that we're training with ground truth, here's all the other things. You can tell that for all of them, they separate. For some of them, they separate better than others. For some of them, they're more or less noisy. And similarly, these also separate if we are going to train with the entropy. Given that the alphas are equal to the negative entropy, they all separate. So this is kind of like a nice sanity check that these might be interesting signals to use for exceptions versus regulars. Does it perform? So now let's look again at the evaluation loss. So using alphas to be equal to the ground truth, the loss, time to convergence, entropy, max or the norm of the hidden layer. What is the loss using the ground truth? So see, the ground truth, and this is split between the regulars in red and the regulars in blue and the exceptions in red. For loss tree weighting in dark and important sampling in light. So yeah, the ground truth does great. Using the loss does okay. Time to convergence does okay. These three do very, very similar because in some sense the max and the norm are just stand-ins for the entropy. But the most important thing is they do way, way better than baseline. This is the naive version where you don't do any reweighting or resampling at all. And these are plus minus one standard deviation. So they absolutely clobber the baseline. The interesting thing is what happens when I change around the beta. So I told you the beta is the inverse temperature parameter of the softmax. And this is why I kind of made a big deal of it earlier. If we have our beta to be very, very low, that means the hidden layer is going to be uniform-ish pretty much for any input. A lot of these do basically as bad as the baseline with no rerating. Conversely, if we make beta to be very, very high, that means the hidden layer is going to be essentially kind of one hot. Then the baseline itself, first of all, is worse. But then using any of these tricks also is not as good as having this intermediate beta. So having an appropriately tuned beta is very important so that the network can exist in both of these the hidden layer can exist in both of these high entropy and low entropy states. And looking at the time, I'm gonna actually just wrap up. So to summarize kind of what I've shown you is I've used this instantiation of the modern Hopfield network proposed by Krotop and Hopfield, and I applied sort of standard machine learning techniques with a very, very, very major caveat of we don't know the sampling probabilities or the loss weights to use. And I've demonstrated that there are a number of pretty viable signals that we can extract from the network itself that can tell us about how to reweight or how to resample. So this says that, or this sort of suggests the hypothesis that there is some sort of introspection mechanism that when you walk around and you look at stuff, you based on introspection, based on the hidden representation, based on how quickly it takes you to converge to the fixed point, which is kind of a stand-in for how easily you can recognize it. So through these introspection mechanisms can sort of tune how aggressively you learn that item, whether you want to memorize it brute force and just sort of slam it into one of the hidden neurons synaptic weights, or you want to gradually learn it in a sort of distributed fashion in the hidden layer. If you're curious, the weird thing that you all hopefully memorized is a sea slug. And I'll stop for questions now. Yes.
Hi. I was wondering if there's any way to pick this adjustment mechanism of the sampling probabilities that wouldn't require you to access the entire hidden layer.
Time to conversions. That's one of the things where you don't need to access the hidden layer at all. You just measure how long it takes for you to get to a stable representation in either the visible or the hidden layer or both. But usually have some sort of timer that says how long did it take you to get there. That's one alternative. Loss is kind of a sketchy one because it requires you holding on to the ground truth and to the output at the same time. But that also doesn't require the hidden layer at all. That just requires the visible layer. I think, I like the hidden layer because it kind of gives you a lot more refinement over the mechanisms and other things to sort of poke at. The other sort of like sketchy thing about having access to the hidden layer is this softmax, right? This softmax requires the entire hidden layer to know about the entire hidden layer to normalize the denominator. I can wave my hands and say that, okay, like maybe there's inhibitory recurrence within the hidden layer, and therefore the entirety of the hidden layer knows about the entirety of the hidden layer. I can also use that to justify the fact that I'm calculating entropy over the hidden layer, or the max of the norm. But yeah, those are kind of the two answers that I have. Yes.
Yeah, I'm curious how robust this effect is, I guess. Like, the thing I have in mind is, let's say we train on your data set, the zeros and ones, and now I show the network at test time a 3, something like that.
Yeah.
Of course, I'm not going to be able to, float to a 3, but can I at least identify that as an exception? like, can I use the entropy to see that this is some new class?
Yeah, so the kind of framework that I'm using, and this is, yeah, I skipped over this, but the framework is you have your data set and you're holding onto that data set. There is no like sense of novelty. Like the entire data set is available, it's just unlabeled. So this is, so you have like some sort of fast learner potentially at the campus that's going to just like store the entire thing. And I'm not modeling that. I can use it. And the goal is like, given this like really big buffer, how do we do the consolidation plus memorization procedure? So then if you have a 3 in that buffer already, then I think everything goes as planned. If you have a pre-trained network and then you show it a 3, then you're kind of in trouble. So you need to have that 3 needs to be like in the context of the entire buffer. Now the other answer is, okay, so suppose you have you don't have a buffer, you see a 3. What you can do is you can take your network and you can feed it random noise and then you can generate a buffer. And then you can rerun this whole procedure. Because now when you feed the network random noise, it's going to converge to whatever fixed points that exist in that network. And it's going to be mostly zeros, and it's going to be a couple of ones. And then it's in the context of that entire buffer, then you're going to be able to see that the 3 is a weird one.
Thanks.
Yeah.
Yes. I guess this is sort of like a follow-up. Is the reason why we use batch gradient descent as opposed to stochastic gradient descent is because we are assuming we're working in like the long-term scheme?
Batch gradient descent and stochastic gradient descent are the same. Like the thing that like the batches are drawn stochastically, and that's what makes it stochastic. I put batch in there just to sort of highlight the fact that we are creating batches.
Oh, sorry, like online.
Oh, like one item at a time.
Yeah.
In principle, yeah, so that's effectively the same thing as using a batch size of 1. I don't see any immediate problems other than simply just like practicality of doing one item at a time is going to take a long time. It might cause instability in the training, potentially. I am not certain about that. But so I suspect that you can just comfortably set batch size to be equal to 1, and it'll be, yeah.
Have you thought about the roles of initial conditions for your network, and have you looked at the distribution of entropys before you do any learning?
Well, so I haven't looked at the distributions, but the entropys before I do any learning are, yeah, it's pretty hard to predict. So the way that I initialize the network before I do any training is like random Gaussian, like the W's drawn from, I think like normalized Gaussian. Normalized, so the standard deviation is like 1 over the number of neurons, the gradient properly nicely. But yeah, the entropy for regulars and factoring is actually the same at initialization. And I haven't considered the scenario of like continual learning where I train it on 1s and 0s, and the next day I train it on 2s and 3s, and that would be like a very, very different initial condition. But starting from random Gaussian initialization, you cannot distinguish them. You can only distinguish them after some training has progressed. That's why this is like slightly miraculous that we in fact can bootstrap it.
So this is like average, right? Like this is the average entropy.
Yeah.
Okay.
Yeah.
And if you don't average, do you have a sense of how wide the spread is?
Oh, I don't know. I should plot the spread. The spread is like big. Like I think the spread is like on the order of like out here. It's not tight. However, on average, it appears to be good enough for it to extract some sort of meaningful information to actually get the ground truth loss to go down, despite using any of these signals that don't have any access to ground truth. Yes.
So I was wondering, basically on the exception, I think, but then the exception ones get weighted more in some, they have a, I guess, a lower entropy. And so you could imagine that at the time they both get the same entropy in the end because they get 3%, as if they had like more items.
So, like, there is no advantage for the network to do this because what, like, in order to store a rare item, what it's doing is effectively it's taking away one neuron from the network and then learning the regular class, the consolidated class, and then putting that neuron back. So you're not getting a hit in capacity by doing this. However, if you now try to store a distributed version of this memorized item, you are taking a hit because now you're taking away capacity from the hidden neurons that will represent the consolidated items. This is only an intuition. Like, there is no, like, I don't have any theorem that states this must be true. And in fact, what you're saying is it might be possible, like, yeah, for a different data set, for a different training regime, that entropy cannot separate them. But for this one, it does seem that it can. Was that your question?
No, I just want to, I understand that at the beginning the entropy is 90%.
At the beginning, the entropy is the same.
Or like at the beginning, I don't know. Like if you, like I guess in a sense it seems like if you re-weight them, in a sense the exception was getting more weight.
Yes.
This is kind of not the same as, you know, it is not the same as having like many of the regular class that we weight less because the regular class is more examples, so they're kind of different in themselves. Maybe.
Yes. Yeah. No, we can turn out that line, but yeah, I think what you're saying is is correct. Yeah, you're upweighting the exceptions, you're downweighting the regulars in effect, so that they are equally important. So you're attempting to replicate this ground truth where you take the average over the second exceptions and the average over the regulars, and then those two averages when you average it.
But the representation somehow in a way that still the exceptional ones have a lower entropy.
Yeah. Yeah.
Okay, let's thank Danil again. And our first speaker is Alexandre coming from Barcelona, talking about working there. That's all.
Hi, so thank you for the invitation. It's a great pleasure to be here at CUNY. First time in New York, so I'm super happy. Yeah, so I work in Barcelona in this brain circuit and behavior lab, and there is a lot of computational people in Barcelona actually. So if you're interested, you can go. We have this community called Maxim. And today I'm going to talk about how you can learn stable working memory representations in a complex task that's called the dual task. Okay, so in our lab, we're very obsessed with a bump attractor. So I'm plotting a bump attractor now. And so this is just a way of representing working memory, which is a persistent activity of neurons after presentation of a stimulus. And as you can see here, the bump is kind of diffusing and drifting, and the location of the bump gets corrupted with time. And this is because working memory is very fragile. So you have this working memory degradation, which is just diffusion of the bump. And another aspect of the fragility of this working memory is how any strong distraction can affect the memory. And well, these are some work that are investigating all these things. And so this idea of having like a working memory that is robust to distraction is something very important to be able to function. And it's been gathering a lot of focus. So for example, in the Romanis and Svoboda lab at Genelia Farm, they developed this simple go-no-go task where it's very fancy because the stimulus is some photostimulation of the somatosensory cortex of the mouse. But basically, either there is stimulation or no stimulation, the mice wait for a given delay of 2 seconds and they need to lick right if there was a stimulus and lick left otherwise. And so they show that you have neurons encoding both lick left, lick right, etc. But the picture is that they go and perturb also with some photostimulation the activity of the neurons in the delay period, and they see that in the naive animals, the perturbation can lead to switching the choice of the animal. So either the animal had the stimulus and decided to lick right, and so you're going to perturb and switch that. So, and what they found is that when the animals really learn the task, they become more robust to the distraction, and they have this dynamical double well attractor picture of this process of the learning of this task, which will be like you start with two wells that are close by with a small barrier, so that any distraction can switch you from one well to the other. And then when you have learned the task, the dynamic of the system changes. Now you have two strong and deep wells that are further apart. So this is more of a dynamical picture of what could happen in the neural activity. Another picture would be something more geometrical, and this is what's been developed by the Bushman lab recently, where they also have a working memory task, but now it's more about how two stimuli can interfere with each other. So here the stimuli, you show two stimuli, then there is a delay, and there is a cue that tells you which stimuli to report. But the important message here is that what they found is that if you look at the neural space and you try to understand geometrically what's happening, they found that when the animals learn the task, these two stimuli, they're stored in orthogonal subspaces in the neural state space. So what do we do with this dual view? Something more dynamical that comes from the learning, or like there's a geometry in the brain, we have a lot of neurons, and eventually everything is stored in orthogonal subspaces. And this is what we wanted to investigate in something more powerful than just a task with a distractor. And this is the dual task. So this is a work in collaboration with the Institute of Neuroscience in Shanghai and the group of Cheng Yuli. And they've developed this very complex task. So you have animals head fixed in a little tube that can receive odors and water from some ports. And the task consists of an olfactory delayed paired association task, which is just a standard working memory task. So you present a sample odor, A or B. There is a delay, and there is presentation of a test odor, C or D. And the animals, they need to learn to match A with C and B with D in order to get a response, a reward, sorry. So if they lick when you present A, then C, they get a reward. Same thing for B and D. And when A goes with D or B goes with C, there is no reward and there is no punishment. So in order to perturb the information of the sample A and B during the delay, we introduce a second task that the animals need to perform, which is just a delayed response task or go-no-go. So there are two new odors, a go odor and a no-go odor, a little delay, a Q odor, and the animals need to lick when the go odor was presented. It looks a bit complex, but hopefully you understood the picture. No, there is never any punishment here.
Okay, but so is there anything active you need to do when they get the no-go order?
They don't need to do anything.
Then they can just lick every time, right?
So I can show you the leaks. Actually, so we have the leaks, and these animals are super smart, and they only leak during Q presentation when there is a go signal, or during the test odor presentation. We check for that because otherwise everything could just be like motor and yeah. Anyway.
The only thing that stops them from licking when you're not supposed to is to say yeah.
Yes. But actually there was a shaping process before they all actually got trained, and which is quite complex. They've been shaped on both tasks separately, and yeah, I mean, these guys are kind of magicians to get animals to perform complex tasks. So anyway, so we can see that the animals, they do learn the task in a few days, so 10 days roughly. And that there is this, so in the task, we have these three types of trials that I will call DPA only with no distraction, dual go when the go stimulus is presented in the go-no-go, and dual no-go, the other one. And they're lived pseudo-randomly during the sessions. And we can look at the performance. So this is the performance of the outer task. So how well you can match sample with test order, okay? And we see that in DPA and Go, you have the same performance for the animals, and the Go trials are more difficult, which is what we would expect intuitively, right? Because it involves getting a reward, so maybe then the animals lose focus or whatever, and also there is this motor action. But these trials are more difficult, and this is what we're trying to understand here. So the data that we have is 2-photon calcium imaging data from the medial prefrontal cortex of mice in 2 different regions. So the prelimbic cortex and the anterior cingulate cortex. We have 5 mice across 6 days of recordings. Depending on the mice, we could track neurons across days, so from 100 to 700 neurons, and we have 96 sessions per day. So that makes the data very sparse, but we can still say something about the population activity. This is just a picture to show you the raw fluorescent signals, and you can see here these are different neurons for different trials. You can see already that different neurons are encoding or selective to the different events of the task. The performance is very similar to what we saw in the batch of animals, which was just doing behavior with the animals having a harder time learning the dual Go trials. And because here it's clear that the first 3 days are more difficult than the performance is different, but not so much in the last 3 days. I'll do this dichotomy between first days, naive animals, and last days, well-trained animals. Okay, so first thing we can do is to check that actually MPFC is kind of encoding the sample memory and keeping it across the delay of the task. And to do that, we can train a linear classifier to classify between A and B. And to get the full picture, we can do that in a cross-temporal decoding manner, which is we're going to train at each time point and test at each other time point. So this is this little schematic there. And so this is the kind of picture that you get with some nice code, usually along the diagonal. So the first row on the first days of training, and the last row is training the decoder on the last days of training. So you see that in these DPA trials, the code is like very robust and very stable, which will be all these nice warm colors. And it gets even more stable when the animals are well trained. But in those dual Go trials, so here and here, we see that the representation of the sample information changes after the distraction. So this is just a F1 score, but you get a very similar picture if you take any kind of metric. It's the F1 score to classify A or B. So what's interesting is that if you look at the early code, so here in here, you see that you lose that in the first days of training in those dual Go trials, and learning makes you recover and stabilize the early representation, so that you keep the same information despite being distracted. And so, okay, so this is basically this off-diagonal score. This is exactly the decoding on the early delay and training on the later delay. And this is the picture that you get. So but it's not super good because the data is sparse, so there is a bit of overfitting, but the trend is kind of similar to what we see in the performance. Okay, so if learning the dual task has to do with having a robust early memory representation of the samples, we can now look at something more interesting than just this low-dimensional representation and take the full population activity and project it on this subspace that we found for the representation of the memory of the sample. And this is what we see here. So this is just the overlap of the activity with the weights of the classifier, which is just the subspace of the representation of the sample. And we see, surprisingly, that when you take the full picture and project it in the low-dimensional subspace, we can't really distinguish between what is a well-trained animal or what is not. So we were a bit puzzled here and went further and looked at what happens if you try to decode the distractor instead. So if you look the same way as the subspace of encoding of the go versus no-go information in the middle delay, so just after the presentation of the go and no-go signals, we see this more interesting picture. When you take the overlap of the population of neurons, what you see is that for go trials, the sample elicits a response in the distractor subspace and along the direction of the go signal. So this is kind of what's there in the first and so naive animals, and that goes away when the animals are well trained. Okay. Yes. So we can look at the amplitude of these little bumps here, and plot them across animals and across the different trial types, and see that actually this is significant. The slope is significantly going down, and that correlates with the false alarms in the task, which is don't leak when it's not a pair trial. Well, that would be correct rejection. So leak when it's not a pair trial. But the idea behind that is why would you store information in the go direction, which is the direction of the response, when the sample is presented, when you're very naive? And this is what we go and investigate in a network model. So I've developed this, based on previous works. I've developed this spiking network model of EI neurons operating in a balanced regime. And we add plasticity to excitatory synapses so that the network can exhibit like multi-stable activity. In order to store memories, we take like a Hopfield-like approach, like, well, a bit similar to what was talked earlier today. But what's interesting about this model is that we respect Dale's law, because what we do is to change the connection probability of the neurons in this specific way. So you have a sparse connectivity with a weak probability of connection P, and we add to that some even weaker probability of connection along the memories, so along Xi S or Xi D. Okay, the dynamics of this network is something like that. So if there was only one memory in the network, what you'll get is this critical pitchfork, which depends on the strength of the low rank structure in the connectivity. Okay, and you go from a baseline non-structure state to a state, a bistable state, where you can have a positive or negative overlap on the direction of the memory. Now, if you add another a second memory, the picture gets a bit more convoluted, and now you have to look at what happens in this sample overlap versus distractor overlap space, and you go from one fixed point to a fixed point at well, this is not exactly zero. This is there is activity in the neurons here because we are in a balanced state, but so what you get from a single fixed point to a fixed point and stable fixed points along a ring attractor. And this is work that's already been investigated by Francesca Mastro Giuseppe and Ostojic, and there is also other papers of this similar dynamics by Manuel Beran, which is now somewhere here, I think. So. But this is the dynamics of the network. So in the network, we have very few parameters in the end. We have the two memories and the two inputs, the sample and distractor inputs, and the only thing that matters is to choose how they covary together. And we can tune the parameters of the model so that the model will fit the data. So in order to observe a response after on the sample overlap, after sample overlap, you need the sample input to covary with the sample memory. So that's this little plus there. I can't see that. This little plus there. And so what we needed to get this early bump in the distractor overlap after sample presentation, which is the one that we see here, it's this little plus here. And you go from that picture to the well-trained picture by adding zeros in this covariance matrix along the diagonal. So the two memories, that's a requirement from the model, needs to be orthogonal when they didn't need to be like that in the first to represent the first days. And you also need an orthogonalization of the sample input with respect to the distractor memory. Okay. So this is just simulations of the network to show that we have kind of good match with the data. Okay. So I just mentioned this orthogonalization of the working memory representations that we need in the model to reproduce the two conditions, naive animals and well-trained. And we check for that in the data. So here I show you the angle between the two memories extracted from the data in one animal. And you see that it goes from 100 to 90 in the first 3-4 days. Okay, and then it stays around 90. There's this glitch here at 5, this outlier, but it kind of correlates with the behavior that we see here in the inset. And this will be the population structure across mice. So we have a significant decrease of the angle between the two memories, and it goes to 90 in the well-trained phase. It's also significant. One other thing is that you see that in the beginning, those two memories, they're not like completely aligned and they're not too far from orthogonal. This is something that I didn't mention, but this is this epsilon here. So in the model, if those two memories, they're aligned, well, you can't get all these interesting dynamics. So this is also a requirement of the model that the two memories, they should start not too far from orthogonal. So now the story becomes interesting because the model tells you that if you have non-orthogonal memories, the dynamics of the system is going to be on a double well attractor, and the wells are going to be aligned across the diagonal, either the positive diagonal or the negative one, depending on the signs of the covariance between the two memories. And you go from that to something orthogonal, which should give you this line attractor dynamics in the model. Obviously, this is true only for infinite networks. And here in my model, we have some noise in the connection. So you get the full line attractor if you initialize different matrices, but that's the picture. So now we have this kind of ring attractor picture in mind, and we can go and try to give some orientation selectivity to these neurons. So to try to find the angle on this ring that we found, for which the neurons are more selective. And we can do that in the model very simply, like that, with this mapping of the angles. And so you see here on the left, we have the network with unordered neurons, and then you see this bump activity when you order the neurons. And you tell me, well, you'll never get that in the data. This is like a prefrontal cortex of a mice. It's not even doing anything visual, right? But so this is a bad trial. This is a single trial of the data that we have. But when you order the neurons based on the memories that you extracted, you kind of see this bumpy activity, and you can label a phase of the bump. And this is a better example. So another order, and here you clearly see this persistent activity and well, this kind of bumpiness of the neural activity. So now we've got the phase of the bump in the neural data, and from that phase, if we assume that what the bump is doing in the data is just diffusing randomly, it's kind of a random walk, we can consider that's a Markov process, compute its transition matrix, get the steady states of the process, and from that, assuming that we have a kind of a Boltzmann distribution of these states, get an idea of the energy landscape behind the dynamics of the phase. And this is what we got from one animal in PRL. So in red, in blue, we have the first days, and in red, the last. And here on the ring, I'm just putting where the wells are and where the height of the barriers are. So we go from a picture where you have two wells that are separated by almost 180 and along a diagonal here, to a situation where the two wells are close by. And well, here the barrier looks a bit smaller, but I mean, we don't have any stats on that. So, okay, this is the same picture in a second animal, and in ACC now. So now we have in ACC these two wells that are also at 180, but not along the diagonal. And when the animals learn the task, the wells, they go down and they're a bit more close by. Okay. So we can go back now to the behavior of the animals and try to see if we can, from this like ring attractor or attractor dynamic interpretation, get some insights into what happened into the behavior. So basically on the ring, the cardinals are task-related events. Okay, so up is go, so north is go, south is no go, east is A and B would be west. And so what happens if you have this interpretation? So when you have attractors like that that are on the diagonal, a push in the B direction followed by some kind of a go signal or not even is likely to send you back to the other side of the ring in a slow manner, but still, and more likely than if you had a push in the other direction. And eventually what you would like is have the system fine-tuned with the two wells at A and B, but this would be too much for the system. So it seems like in the data that we have, the solution that the brain finds is to push those wells in the direction of the no-go, of the no response, so that whenever you get a push now from one of these attractors up, you keep on the same side of the ring, so you're not losing the A versus B information. Okay, so from that we can check whether in PRL there was this imbalance between the two samples. Yes?
When you on the mouse parse these two tasks, right? And first, like there's a bistable fork, right? And then like there's no task, it forms like this ring. But like mouse never has one memory, two memories, right? So,
Yes, yes.
How does, like, I didn't really understand how it works. Like, how can you even start with a bistable if there's so many memories already in your brain, right?
Yeah, but this is a simplified model, and you know you can add, you could add like a large number of memories, but
Did you get bistable state in experiments, right?
yes.
So how do you think that works?
Well, I think if you add more rank to the structure, so first these are the task. So these are the events that are important to perform the task. Okay.
Do you think that these attractors form of like current like memories that are like, so the active memories are selected in this kind of circle?
Well, this is like really an interpretation of what's happening in the data. Obviously, the picture is more complicated, and if it's not a ring, it's probably like a high-dimensional sphere or something like that. If you have, I didn't do the calculations to see what kind of structure you get. I guess with 3 you'd get torus, and if you go to n memories, but there should be some kind of a symmetry, and you know, it's always hard to go from 2 to n something. Yeah. Okay. So with this picture, what we get is that if the attractors are not symmetrical along the y-axis, there should be an imbalance in the responses, in the correct responses in the different types of samples. And this is what we checked, and we see that in PRL you have this imbalance, and it matches our interpretation of how this ring attractor dynamics should explain the learning of this task. In ACC, where these two wells that we derived were kind of symmetric along the y-axis, we don't see this imbalance. So I hope I've convinced you that there might be more to stabilizing working memory in prefrontal cortex of the mice than just a double well attractor or this orthogonalization. Here we propose a framework where kind of there is a conjunction of the two effects, and we'll see if we can get even more insights in the future about that. So in conclusion, I've showed you that MPFC displays some neural dynamics that is closely linked to the behavior of the learning of the dual task, that this learning leads to the orthogonalization of the two memories, the sample and distractor memory, that in order to have distractor resistant code, well, there is this kind of double well dynamics on the ring that pushes the wells towards the no-go direction. And this is kind of a dynamical tuning. It requires both orthogonalization and this kind of ring structure. And with that, I want to thank my colleagues in Shanghai, my supervisor Albert Conta, and the rest of the people in my lab in Barcelona. And thank you for the invitation, and thank you all for listening to me.
And you said arbitrary units. So I understand why I don't know where zero is. But if I think of the energy as being minus the logarithm of probability, then the scale of variation should be mean.
Yes, I think it has to do with the units of the fluorescence. Of the fluorescence? No? No. So you mean the units, or you mean?
You have this picture of, if I take the e to the minus this energy, I should get a probability, right?
Yes.
So there's a problem with normalization. That tells me I don't know where zero to the energy.
Well, yeah, here I removed the minimum of the landscape. Yeah.
But if I take this at face value, the modulation of the probability is only 1%.
Yeah. Yeah. So here the energy is so I'm also normalizing by yeah, I'm dividing by the mean energy. So that might change that's rescaling of it doesn't affect the first and last curves. They're still on the same scale, but it's just so that we could have similar comparisons between animals. Unfortunately, I changed these plots yesterday, and now I don't understand why there is this big scale difference.
Okay, so our next speaker is Antonio Fontanella, coming close from the University of Arkansas. Going to talk to us about critical dynamics.
Thank you. Okay. Critical dynamics in the subspace for spontaneous behavior. I'm Antonio Fontanelli. I'm really thrilled to be here. Thank you so much. Also, my first time in New York. I'm really happy and excited. Thank you for the invitation. And also, I'd like to say thank you initially to my team. This work will not be possible without the support of these people. John Suter, Galpam, Norman, and Dr. Shu, my advisor. And okay, a long-standing question in systems neuroscience is, what's the dynamical regime of the cerebral cortex during awake state, right? And I'll just show you some maybe 20 seconds of recording of an awake mouse on top of a wheel. And for example, 200 neurons here. Here's just the population activity. And I will ask you, what do you think that is this regime? Is it synchronized, desynchronized, and what do you think about it, right? We are from the camp of criticality. We believe, our thought is that the cortex is right at the boundary between uncorrelated and a more highly coordinated regimes, right? So it's right in the boundary between those two extremes of just arguing by hands here what is synchronized and desynchronized, right? And also, I think that it's important to mention that criticality is, we can see, for example, a high diversity of time scales in a system close to a critical point, and the levels of correlation are moderate levels of correlation that you have here. And also, the people argue that the first people that start criticality, at least from this approach, right? There is another approach to study criticality in the cortex. That maybe it's interesting to promote some optimal transmission capacity, optimal sensitivity, and dynamic range. and maximize the information capacity, right? And so those are thoughts from the beginning of this way to look at the dynamics of the cortex. And it started in 2003 with bags and plants, where they recorded the activity of slices, basically in vitro preparations, just brain in a dish, right? And they recorded the activity of those neurons, and they proposed that if you split the activity of the neurons in some boxes, this one is delta t equals 4 milliseconds, you will find some epochs of no activity and epochs of continuous activity, right? So, and they gave the name of this period here of neuronal avalanche, and it's characterized by a size and by a duration. The size is basically the amount of activity between silent periods, right? And the duration is just the number of consecutive beans that you have, right? And they also most interesting that they found that those quantities, size and duration, are you can fit a power law model to those distribution, right? With this, for example, for this the size of the avalanche, we have this power law distribution characterized by this exponent minus tau, and the duration minus tau t. And also by the end of the first decade of this hypothesis, Friedman and colleagues proposed another scaling relation, but this time connecting the mean size of the neuronal avalanche and the duration, connecting through this 1 over sigma nu z exponent. And also they found that those three independent exponents, 1 over sigma nu z and tau t and tau, are connected through this relationship that the people call it crackling noise relationship. So basically, this is the framework of criticality from the cortex from this point of view. But okay, all those results are models or brain in a dish, right? It's just in vitro observations. And what about what about the awake state, when the cortex more demand coordination, when it's more relevant, I think. And when the people start to look at the awake state using, for example, spike data, there is the scale of milliseconds, some controversial results appear. For example, Ribeiro and colleagues 2010 found that the distribution of size are really curved, really close to the surrogate, right? And also Han in 2017 found the same curve and really short power law range. And it cast some doubt about this hypothesis, right? That the cortex operates close to a critical point from this point of view, of course. And here is a summary of the research, the research that found positive signatures of criticality, those white background here, and the yellow ones are those ones in awake, of course, that using spike data. This is relevant here, awakening spike data. And those yellow ones here are those ones that found negative reports. But if you look here, there is a kind of pattern. Those ones that you found negative reports, they used really short time beams. Remember that this little box that you split your data and calculate the estimate the population activity and look for size and duration along this population activity. But those ones that reported positive results are those ones that used a coarse time beam, right? Here they used from 1 to 20 milliseconds, and here